Hi everyone, I'm super happy to announce that this is my all-in-one Human Biology IGCSE talk through. So I'm going to be taking you through every specification point, hopefully giving you lots of helpful tips along the way. Remember, if you like the sort of points I'm making, I've written a revision guide specifically for your exam board. It's available to purchase on the website. But we're going to get stuck in, number one, with microscopy. So let's start by talking about how we set up a light microscope in order to view a sample. So first of all, you want to affix your small sample onto a microscope slide, add water or a dye depending on what you want to do with it, but the all-important cover slip also needs to be added. Then you're going to pop that slide onto the stage of the microscope, turn the lamp on so that the light shines through the slide, and you're going to start with your lowest objective lens in order to start bringing that slide into focus. Do make sure that the slide doesn't touch the lens of the microscope. Once that slide's in focus, you'll move to a higher objective lens and you keep moving that coarse focus dial to bring that slide into focus. Now, the two main microscopes are the light microscope and the electron microscope. So let's talk about the relative advantages and disadvantages of each. So the light microscope, first of all, it is much cheaper to buy. It does not require specialist training. If you're at school, you might have actually used one yourself. You'll realize that they're pretty easy to use. Major advantage is that your specimen can be live and it therefore enables you to see its true color. The major disadvantages with light microscopes are both their magnification and resolving power. So first of all, let's consider, what does the term magnification means? Well, it's the number of times larger the image size is compared with the object size. The resolution is the ability to distinguish between two separate points on an image. So the higher the resolution, the clearer the image. So going back to our light microscope, unfortunately, it can only magnify up to 2,000 times greater than the original object. The resolving power isn't the best. It can only distinguish between 2 points 200 nanometers apart. You'll often be asked to calculate magnification in the exam, so remember the equation magnification equals image size divided by object size. It is important that your units align, and they do like to try and catch you out in the exam. It's up to you which unit you use, but if you go with millimeters, everything needs to be in millimeters. If you go with micrometers, everything needs to be in micrometers, and you may need to convert between the two in order to ensure those units align. To get from millimeters to micrometers, you're going to multiply by 1,000. From micrometers to millimeters, you're going to divide by 1,000. Now moving on to the electron microscope. Here, the disadvantage is that they're hideously expensive. You need very specialist training, and the sample is going to be dead. The image you produce is black and white, unless you colour it up afterwards in post-production. But the major advantage here with the electron microscope is that you can magnify up to 2 million times and the resolving power is 0.2 nanometers. That's amazing. It means you can distinguish between 2 points very close together. This enables us to see cell structures that previously we were unable to observe. Moving on to cell structure, so looking at the organelles we find in eukaryotic cells. Remember, eukaryotes are cells containing membrane-bound organelles such as the nucleus and the mitochondria. What's the role of the nucleus? Well, it controls the activities of the cell. It contains the cell's DNA, the genetic material. It's also where you'll find the chromosomes. So the chromosomes are strands of DNA. Remember that a section of DNA which codes for a particular protein is known as a gene. Moving on to the mitochondria, this is the site of aerobic respiration. ATP is made and energy is released. The endoplasmic reticulum contains ribosomes, which are the site of protein synthesis. You've got the cytoplasm, where chemical reactions take place, and the cell membrane, which allows the entry and exit of substances into and out of the cell. Let's now consider DNA. So first of all, the structure of DNA. DNA is a double helix. That means it consists of two strands. The strands' backbones are made out of sugar and phosphates, and they're joined by complementary bases. Remember that adenine always binds to thymine and cytosine always binds to guanine. These bases are joined by hydrogen bonds. A nucleotide consists of three subunits. It's the phosphate, the sugar, and a base. Remember that the sugar in DNA is deoxyribose. DNA is a comparatively long molecule, especially when you compare it to RNA, which is much shorter. DNA replication occurs before mitosis. Remember that mitosis is a type of cell division. DNA replication simply means producing new DNA from old DNA. Now that new DNA will consist of one of the old strands from the original DNA molecule and one new strand which is formed from the original template. The DNA produced is genetically identical to the original parent DNA. Let's go into further detail how DNA replication actually takes place. So first of all, the hydrogen bonds break and the DNA strands separate. Each strand acts as a template. DNA polymerase is a type of enzyme and it's responsible for adding nucleotides to the exposed bases. Complementary base pairing of nucleotides occurs and two identical DNA molecules are produced consisting of one new strand and one old parent strand. Now we'll move on to RNA. Right, we've already said that RNA is a shorter molecule compared with DNA. The sugar in RNA is ribose, whereas in DNA it's deoxyribose. DNA, remember, was double-stranded. RNA is in fact single-stranded. The bases vary slightly in RNA. You still have adenine, you still have guanine and cytosine. The difference here is that thymine is replaced with uracil. Broadly speaking, the role of RNA is to take information from DNA in the nucleus to the ribosome, which fundamentally allows protein synthesis to take place. Moving on to mutations now. So mutation is a spontaneous, rare and random change in the DNA of a person. Certain chemicals may increase the likelihood of a mutation taking place, such as carcinogens, such as tar, ionizing radiation, UV light, exposure to X-rays and gamma rays can all increase the incidence of mutation occurring. A mutation in DNA can lead to a change in the base sequence. This alters the amino acid sequence, meaning that a different protein is made, potentially with a new phenotype. The effect that the mutation has totally depends on the type of mutation. So if you're adding extra bases as part of your DNA mutation, that will clearly change the amino acid sequence in its entirety. Sometimes when you simply have a substitution of bases, you actually end up with the same amino acids being brought to the ribosome. We'll talk a lot more about that very shortly when we go into the detailed explanation as to how protein synthesis works. So protein synthesis is split into two stages: transcription and translation. Transcription takes place first of all inside the nucleus. Here the hydrogen bonds holding together those complementary bases break. The DNA unwinds, exposing its bases. There are free RNA nucleotides present in the nucleus which pair up with the exposed DNA bases. This enables a single-stranded mRNA molecule to be formed. That mRNA now leaves the nucleus and binds to the ribosomes in the cytoplasm. It's important that you're aware that three bases on mRNA is known as a codon. The codons are complementary to anticodons, which are found on one end of a tRNA molecule. At the opposite end of the tRNA is an amino acid. So effectively, the order of codons dictates the order in which the amino acids are brought to the ribosome. The amino acids join together to form a polypeptide, which folds into a complex 3D shape to form the end protein. Remember that in terms of the possible number of codons, the way in which you work that out is by contemplating the fact that 3 bases is responsible for each amino acid. So if you do 4 cubed, that tells you that there's a potential 64 codons. Now we're moving on to genetic engineering. So what is genetic engineering first of all? When genes are altered in a microorganism to code for a desired product. One of the most famous examples here is modifying genes in a bacterial cell so that it produces the hormone insulin, which can be used to treat diabetes type 1. You need to know the names of 2 very specific enzymes. Number 1 is the restriction enzyme used to cut DNA. Number 2 is the ligase enzyme used to join the cut ends of DNA back together. Another good word to know the meaning of is the term vector. A vector is used to transfer biological material from one organism to another. A good example is that bacterial plasmid I just described in insulin manufacture. Another example is the mosquito, which transfers the plasmodium to the human host. A plasmid, remember, is a small circle of DNA found in bacterial cells. And now we're ready to describe in detail how bacterial cells can be genetically engineered to produce human insulin. So remember, human insulin is needed to lower our blood sugar levels after we've eaten a meal. It converts glucose to glycogen. For people with type 1 diabetes, they're unable to make insulin. Historically, they used to use pig insulin, but for all kinds of reasons such as ethics, the fact that the human would reject that pig insulin, it was deemed an unsuitable method. Genetic engineering provided a way in which we could make huge amounts of insulin without the need of injuring any pigs, and it would be perfectly matched for the human recipient. So, number one, a restriction enzyme is used to cut the human insulin gene from a healthy person. The same restriction enzyme is used to cut open the bacterial plasmid. A ligase enzyme is used to seal that insulin gene into the bacterial plasmid, and the sticky ends, which are exposed ends on the DNA, are sealed together. They are complementary to each other. Because the plasmid now contains human DNA, it's known as a recombinant plasmid, and as I previously mentioned, the bacteria is acting as a vector here. The bacteria is then placed in a fermenter, which is a large vessel with optimum conditions to ensure that that bacteria replicates as quickly as possible to make huge quantities of human insulin. That insulin is then purified and is ready for use. As well as genetically modifying bacteria to produce hormones like insulin, it's also possible to genetically modify plants. So now let's run through a specific example. So for example, say we want to genetically modify a plant so that it produces more beta-carotene. Remember, beta-carotene is a precursor of vitamin A, and it helps our eyesight and enables us to see more clearly in low light conditions. Hence the expression, carrots help you see in the dark. Carrots are full of beta-carotene, so therefore good for your vision. So how do we genetically modify a plant so that it can produce beta-carotene? Well, first of all, we need to use a restriction enzyme to open up that bacterial plasmid, and the bacteria we use is known as an agrobacterium. Restriction enzymes are also used to isolate the desired gene in the target DNA. We know that sticky ends will be produced because that will enable the ligase enzyme to seal the desired gene into the bacterial plasmid. And again, we have what's known as a recombinant plasmid. The recombinant plasmid is inserted into the agrobacterium bacteria, and leaf discs from the plant are treated with the recombinant agrobacterium. The leaf discs are cultured in a nutrient-rich medium in sterile conditions, and any plants that grow will have that desired gene, and in this way they're known as transgenic plants because they contain a gene from another species. So you might want to know, why is agrobacterium used here? Because agrobacterium is often found freely in soil, and it is known to introduce plasmids into plant material, making it the perfect bacteria for this process. So we've already described that using genetically modified plants can increase their nutritional value, such as this example here we said, which meant that more beta-carotene was made. Other reasons we might genetically modify plants is to increase their shelf life, to make them more resistant to frost, to make them resistant to herbicides, weed killers, so that when the farmer adds weed killers to his crops, you're not going to kill the actual crop that you're trying to grow. It's even been known that tobacco plants have been modified to produce hepatitis B antigens. This has potential use in creating a vaccine in order to prevent hepatitis B infection. So now we're going to outline the advantages and disadvantages of genetically modifying plants. So the main advantage is that you might create higher yields because of reduced competition from weeds, as well as reduced damage by pests. It's possible that crops can be grown in more extreme environments, meaning that there's less famine due to more crops being grown. Tied in with one of the points I've already made, genetically modified plants mean that less herbicides and less fertilizers need applying, which makes it cheaper and easier for any farmers, and obviously much better for the environment. And as I've already previously mentioned, genetically modified plants increase the nutritional value to humans. They have a more complete combination of lipids, proteins, vitamins, minerals, and I've mentioned beta-carotene on several occasions. But what are the disadvantages of genetically modified plants? Well, they could cross-pollinate with weeds, creating super weeds which are hard to kill using herbicides. They tend to be expensive, and you find that poor farmers can't afford genetically modified seeds. And it also reduces biodiversity by decreasing the species of weeds and pests, meaning there's less food available for consumers higher up the food chain. Now we're moving on to mitosis and stem cells. So to begin with, mitosis is a type of cell division used in the growth, replacement and repair of cells. Because of this, the type of daughter cells it produces are genetically identical to the parent cell. You'll find that mitosis is also used in asexual reproduction, which remember is when there's one parent. unlike sexual reproduction where you need 2 parents. The cell cycle is the name given for a series of stages where body cells divide. It's made up of 3 distinct parts: interphase, mitosis, and cell division. Interphase is when the cells grow bigger and the DNA is replicated. You can think about interphase as the preparation step, so it's when the cells are getting ready to divide. So what do they need to do to produce more cells? Well, obviously you'll need more DNA. So in interphase, DNA is replicated. You need more organelles, so more organelles are made, and the cell itself grows bigger. The second stage of the cell cycle is mitosis, and we'll talk about the 4 distinct parts of mitosis very shortly. And then lastly, cytokinesis occurs, which is when cell division takes place and you find your 2 genetically identical diploid daughter cells are made. Remember, the term diploid refers to the number of sets of chromosomes present in a cell. A diploid cell has 2 sets of chromosomes. A haploid cell contains just 1 set. So the 4 stages of mitosis can be split into the following stages. We're going to use the letters PMAT to help us remember. So the first stage is prophase, and this is when our chromosomes condense. They shorten and thicken and become more visible. The second stage, MAT, is metaphase. This is where the condensed chromosomes line up at the equator of the cell. Anaphase, this is when chromatids start to move to opposite poles of the cell. And then finally telophase, and that's when the nuclear membrane reforms. Moving on to differentiation. So differentiation is the term used to describe when cells become specialized. So for example, when they become nerve cells. A stem cell is a cell which has the ability to divide many times whilst remaining unspecialized, undifferentiated. There are 2 types of animal stem cell you need to be aware of. The first one is embryonic, the second one is the adult stem cell. As the name suggests, embryonic stem cells are found in embryos, particularly the early stage of development of embryos. They have the ability to differentiate or specialize into any cell types. So immediately you can see the added advantages of an embryonic stem cell. It can become any type of cell you want. For example, a bone cell, a hair cell, a nerve cell, a muscle cell. Moving on to adult stem cells, they're found in very specific places, namely the bone marrow, the skin, and the lining of the small intestine. Although they have the ability, like all stem cells, to divide many times, unfortunately they can only become one specific type of cell. So for example, stem cells found in the bone marrow can only become blood cells. Stem cells have lots of uses in the treatment of disease, and this is known as stem cell therapy. A good example of stem cell therapy is in the treatment of leukemia, which is a type of blood cancer. So when the person is treated with both chemotherapy and radiotherapy, you find that both the cancerous cells and healthy cells are destroyed. So that's great, you've removed the cancer, but a person obviously really needs their white blood cells. So stem cell therapy involves introducing stem cells which are able to replicate and differentiate in order to replace those lost cells. Parkinson's disease is another good example of a disease that can be treated using stem cell therapy. In Parkinson's disease, nerve cells are destroyed, so it therefore makes sense that stem cells can be used to replace those damaged nerve cells. Ideally speaking, you'd use a person's own stem cells in stem cell therapy. The advantage here is that they're readily available. You have a complete tissue match, which means no chance of rejection by your own cells. And from an ethical point of view, it's great because there's no need to use embryonic stem cells. So if we consider the ethics now associated with embryonic stem cells, these have to come from aborted or miscarried fetuses. So obviously huge ethical concerns there. But the advantage of embryonic stem cells, as I've already pointed out, is their ability to specialize into any cell type. This puts them at a massive advantage compared with an adult stem cell, where I've already said there's only very specific places where you find adult stem cells. So this is why when people have horse riding accidents and they paralyze themselves from the neck down, you tend to find they remain paralyzed for life, and that's because you don't have stem cells in your spine. However, embryonic stem cell therapy is a way of potentially making more nerve cells in order to repair those damaged nerves in your spine. And I'm just going to drop a table from my revision guide into this video so you can look at the various advantages and disadvantages associated with the use of stem cells. Moving on to cells, tissues, and organs. So we're going to start by listing a whole lot of definitions. Remember, you're going to use the same scaffold for all these definitions. So if we start with a cell, it's a group of similar organelles working together to perform a particular function. And it is that series of words we're going to use as our scaffold, but just changing a word each time as we move through the definitions. So if we take a tissue now, well, that is a group of similar cells working together to perform a particular function. An organ is a group of similar tissues working together to perform the same function. An organ system, well, it's a group of similar organs working together to perform the same function. So if we list some examples of organ system, could be the digestive system, which is made up of a group of similar organs, so stomach, esophagus, small intestine, large intestine, pancreas. Another type of organ system could be the reproductive system, that consists in a female of ovaries, fallopian tube, uterus, cervix, vagina. Now we're going to discuss the structure of bone. So bone is a tissue consisting of cells known as osteocytes, which secrete a hard substance containing calcium salts. It forms a rigid material which resists being compressed, and the osteocytes and the presence of protein fibers stop that bone from being too brittle, so easily snap. There are 3 types of muscle you need to be aware of: voluntary, involuntary, and cardiac. When we talk about voluntary muscle, we're really referring to skeletal muscle. When we talk about involuntary muscle, we're talking about smooth muscle, and then cardiac muscle is associated with the heart. So going into further detail, starting with that voluntary striated muscle, you'll expect to see many nuclei per cell. There won't be any branching, and it will be striped or striated in appearance. Its function is that it rapidly contracts in order to move our bones, and it's controlled by our brain. Moving on to involuntary smooth muscle. Again, you'll find that it's not branched, but this time there's a single nuclei per cell. It's tapered at the ends of each cell, and it's non-striated. It doesn't have striping. In terms of the presence of this smooth involuntary muscle, you'll find it in the walls of our gut, so our small intestine, and our blood vessels, and as the name suggests, it is not under voluntary control by the brain. And then finally, cardiac muscle. This is associated with the heart. Cardiac muscle is striated, it is striped, it has many nuclei per cell, and it's branched, which enables it to form a mesh-like structure. Cardiac muscle is only found in the heart, and that cardiac muscle contracts rhythmically and constantly throughout your lifetime without tiring. Remember, your heart muscle is pumping all day long, 24 hours a day for the entirety of your life. Really, it's the hardest working muscle in your body. Moving on to the structure of blood. So blood consists of a jelly-like substance known as plasma, which is responsible for transporting hormones, carbon dioxide and urea around our body. You're going to have red blood cells inside that blood, which, remember, transport oxygen. White blood cells consist of both phagocytes and lymphocytes, and they form part of the body's defense system against pathogens, which are microorganisms which cause disease. And then platelets are small fragments of cell which clot the blood at the site of a wound. 2 types of tissue you need a bit more information on are the epithelial tissues. There are 2 types, the ciliated epithelium and the squamous epithelium. So the ciliated epithelium are found lining your trachea. They consist of cells which have small projections on them, hairs, and these waft backwards and forwards in order to remove mucus laden with bacteria from your trachea, so that none of this dust and bacteria enters your lungs, where it could cause a great deal of harm. The squamous epithelium cells line your cheeks, and as such they're flattened in shape. Moving on to more specialized cells, so the sperm cell. The function of a sperm cell is obviously to fertilize an egg. So in order to do that, first of all, it's important that you're aware that the nucleus of a sperm cell contains a haploid number of chromosomes. That means it contains 1 set of chromosomes, so that when it fertilizes the egg, which also contains a haploid number of chromosomes, you end up with that initial cell, the zygote, which contains your full complement of chromosomes, a diploid number. In order for the sperm to carry out its function properly, it needs a whiplash tail to enable it to swim. It needs a midsection which is jam-packed full of mitochondria, which release that energy needed to enable that sperm to swim. And at the tip of its head, it has an acrosome which contains digestive enzymes that help break down the outer membrane of the egg. Moving on to the egg, I've already talked about the haploid number of chromosomes. The egg is much larger than the sperm. It contains a lot of cytoplasm, which provides the energy and materials needed to help the zygote divide to form the embryo. It has an outer jelly coat which hardens the moment a single sperm cell fertilizes it, and that's in order to prevent multiple sperm fertilizing the same egg. Moving on to biological molecules. So what elements are lipids made up of? Remember, lipids are fats. So lipids contain carbon, hydrogen and oxygen. A lipid molecule is arranged in the following way. It's made up of 3 fatty acids and a glycerol. To test for the presence of lipids, you want to add ethanol and water to your sample, give it a good shake, and if lipid is present, you'll see a milky white emulsion. Moving on to proteins now, you'll get the carbon, hydrogen, oxygen elements, but in addition, you'll also find that there's nitrogen and sometimes even sulfur. Proteins are made up of long chains of amino acids. In order to test for the presence of protein, you want to add bioarray reagent. If there's no protein, you'll see a blue colour. If protein is present, you'll see a purple colour. Some good examples of proteins found within the human body include collagen and keratin found in the skin, all enzymes, and hemoglobin found in red blood cells. That hemoglobin, remember, binds to oxygen. Moving on to carbohydrates. Carbohydrates contain only carbon, hydrogen, and oxygen. A good example of a carbohydrate is glucose. Remember, to test for glucose, you add Benedict's reagent to your sample and you need to heat it in a water bath. You'll see a range of potential colours. Blue indicates no glucose is present, whereas brick red indicates that lots of glucose is present, but you could also get a range of colours. So any colour that appears nearer the blue end indicates low glucose presence. Anything that appears near the redder end, so orange, yellow, indicates a higher glucose presence. Remember that glucose is an example of a reducing sugar. Storage compounds of carbohydrates include glycogen and starch. Remember, these are long chains made up of simple sugars like glucose. In order to test for insoluble starch, this time we're going to add iodine. If no starch is present, you'll see a brown colour. If starch is present, you'll see a blue-black colour. Now we're going to talk about the test for vitamin C. This time we're adding DC pip dye. Here you'll add your vitamin C drop by drop and you'll see a colour change from dark blue to colourless. The fewer drops needed to turn that dye colourless indicates that more vitamin C is present. Now we're going to describe an experiment to investigate the energy content of food. So taking, for example, a piece of popcorn. The first thing you need to do is measure the mass of the popcorn using a balance. Add 50 centimeters cubed of water to a boiling tube and measure its initial temperature using a thermometer. You want to set up your clamp stand to allow you to hold that boiling tube. Then you're going to set the popcorn alight. Make sure that popcorn's been placed on a mounted needle, as that will make it easier to hold. So the popcorn starts burning. Immediately you want to hold it underneath the boiling tube because the heat coming off that popcorn will raise the temperature of the water. Do make sure that that popcorn burns entirely. You want complete combustion to take place. That means burning in a plentiful supply of oxygen. And then at the end of the experiment, you want to take the final temperature of that water. You'll then be able to calculate a temperature difference. And then to calculate the energy content, remember we use the equation Q equals MC delta T, where M is the mass of water being heated, C is the specific heat capacity of water, which is 4.2, and delta T is your change in temperature that you've just calculated. Now we're moving on to enzymes. So first of all, defining the term metabolism. This is the rate at which chemical reactions take place in our body. The role of enzymes within metabolic reactions is that they act as catalysts. They are biological catalysts which speed up the rate of a reaction without being used up, and they're chemically unchanged. What distinguishes enzymes from other catalysts is this use of the word biological. They're biological because enzymes are made out of protein. Focusing in on some key enzymatic terms. So what is the substrate? That is the substance upon which the enzyme acts. The active site is the special site on the enzyme where the substrate binds. When that substrate binds, you have what's known as an enzyme substrate complex. And then once the enzyme's finished its work, the substrate is released as a product. Optimum is a word used to describe when the enzyme is working at its highest rate. And then another key term is the word denature. Remember, if the temperature is too high or the pH alters too significantly, the enzyme denatures, which means that its active site changes shape and the substrate no longer binds. Remember that enzymes are highly specific, so each enzyme will only fit one type of substrate. It's highly complementary. The substrate shape is complementary to that of the enzyme's active site. If we now consider the effect of temperature on enzyme activity, as you increase the temperature initially, the enzymes and substrate molecules have more kinetic energy. This means that collisions between the two occur more frequently, so you end up with more enzyme-substrate complexes being formed. The enzyme's working at its highest rate here, and we call this the optimum temperature. If the temperature increases above that, what happens is the enzyme's active site is altered, the enzyme is denatured, meaning that the substrate no longer fits. Additionally, enzymes have optimal pHs, such as the enzyme protease found in the stomach. Its optimum pH is going to be a very low pH because it needs a highly acidic environment around pH 2. So if you increase that pH further, the enzyme denatures and its active site changes shape. Other factors which alter enzyme-controlled reactions include substrate concentration. So if you think about it, it makes sense that as you increase the substrate concentration, the rate of the enzyme-controlled reaction increases because there is more substrate molecules to bind to the enzymes. However, you reach a point where further increase in substrate numbers has no effect on that enzyme activity. Why is that? Because you've only got a finite number of enzymes, so therefore increasing that substrate concentration further has no effect on the rate of the enzyme-controlled reaction. And so we can say here that the enzyme concentration has become the limiting factor. Moving on to inhibitors. So an inhibitor is a substance which reduces enzyme activity. There are two types of inhibitor. Number one is a competitive inhibitor. Number two is a non-competitive inhibitor. So we'll start by looking at competitive inhibitors. So by definition, a competitive inhibitor binds to the enzyme's active site instead of the substrate molecule. It makes sense that that inhibitor is both chemically and structurally similar to the substrate molecule, as this is what enables it to bind to the enzyme's active site. Once that competitive inhibitor binds to the active site, it obviously prevents the substrate molecules binding. Sorry about the seagulls. And one way in which we can reduce the effect of a competitive inhibitor is by simply increasing the concentration of substrate molecules. Moving on to non-competitive inhibitors, this is a substance which binds to the enzyme in a place which is not the enzyme's active site. It is elsewhere on the enzyme. Because of its binding, what happens is it causes a conformational change in the enzyme's active site. So it alters the shape of the enzyme's active site, meaning that the substrate molecules can no longer bind. Because the non-competitive inhibitor binds elsewhere on the enzyme, it does mean that increasing the concentration of substrate has no effect on increasing the rate of reaction, quite simply because substrates and the non-competitive inhibitor are not binding to the same place. I talk a lot more about the practicals in my revision guide, which remember is available in the online shop, but do just remember, if you're looking at an enzyme-controlled reaction, something like following the progress of the enzyme amylase is a good thing to test experimentally. Why? Because amylase breaks down starch into maltose or glucose, and so you can easily see by using iodine when that amylase has worked, because it means that the amylase breaks down the starch, which turns iodine blue-black, into glucose, which means that you get a negative result with iodine, so you'll see a brown colour. Now let's talk about lactose-free milk. Now you often find that people are lactose intolerant. It means that if they drink milk containing the sugar lactose, they're prone to have an upset stomach, so show symptoms of diarrhea, cramping and nausea. The symptoms of lactose intolerance, such as diarrhea, are caused by water leaving cells and moving into the gut, and the diarrhea means that you have poor uptake of vitamin D and calcium ions, both of which are essential for good health. We can use immobilized enzymes in order to produce lactose-free milk, and it works in the following way. So lactase enzymes, remember enzymes end in A's, are attached to alginate beads. The lactose-containing milk is passed over these beads, and the lactase breaks down the disaccharide lactose into the monosaccharides glucose and galactose, so milk passes out free of lactose. We also like to use immobilized enzymes in order to convert the disaccharide sucrose into the monosaccharide fructose and glucose. The enzyme we need to immobilize in this case is known as invertase, which is made by yeast cells. The invertase is immobilized on beads, and then as the sucrose passes over those beads, it gets broken down into glucose and fructose. Did you know that immobilized enzymes are also used in the diagnosis of diabetes? Now, people suffering from diabetes have abnormally high levels of glucose in their blood and urine. So the test strips produced are used to prove the presence of that high concentration of glucose, and they work in the following way. The test strips use two immobilized enzymes. The first one is known as glucose oxidase, which causes the oxidation of glucose to hydrogen peroxide and gluconic acid. The second enzyme is peroxidase, which catalyzes the oxidation of a colorless organic substance into its colored form by hydrogen peroxide, and the amount of coloured substance is a direct measurement of the quantity or concentration of glucose in the urine or the blood. So why do we use immobilised enzymes? Why do we like attaching enzymes to alginate beads, for example? Well, number one, it's because those enzymes are reusable. They don't get used up, as we know is common with all enzymes, and they can be reused again and again. The product produced is pure. The enzymes are produced in a way which enables them to withstand large changes in temperature and pH. They're not liable to denature, another essential property when used in industry. And there's a large surface area created by the use of beads, which therefore increases the rate of reaction. Moving on to the movement of substances into and out of the cell. So there are 3 different types of transport we need to consider. Number 1, diffusion, 2, active transport, and 3, osmosis. So diffusion is the simplest. It is simply the net movement of a substance from an area of high concentration to an area of low concentration. This is a passive process and does not require the input of energy or ATP. You could be asked the factors which increase the rate of diffusion or affect the rate of diffusion. So the steepness of the concentration gradient. The steeper the concentration gradient, the faster the rate of diffusion. Surface area also plays a part. The larger the surface area, the faster the rate of diffusion. A higher temperature will increase diffusion because the molecules have greater kinetic energy. A shorter diffusion distance means that molecules have less far to travel, again, so you'll have a higher rate of diffusion. Unlike diffusion, active transport requires an input of energy. So if we look at its definition, it's the net movement of molecules from an area of low concentration this time to an area of high concentration against a concentration gradient, therefore requiring ATP or energy. Active transport is vital in living organisms because it enables the movement of molecules against the concentration gradient. A good example of this is the uptake of glucose in the small intestine. This occurs by active transport. Moving on to water now. So the movement of water molecules is known as osmosis. So our definition of osmosis is as follows: the net movement of water molecules from an area of high water potential to an area of low water potential across a partially permeable membrane. So osmosis is similar to diffusion in that we're going down our concentration gradient, but crucially it involves the movement of water molecules and requires a partially permeable membrane. A solution which has a high water potential just means that it contains a large number of water molecules. It is in essence dilute. A low water potential solution is one with few water molecules, so could be quite high in solutes, and therefore is what we would describe as a concentrated solution. Remember, it's important that we can move water around in living organisms because water is a valuable solvent. It supports many chemical reactions that take place in our cells, and it enables dissolved substances to be moved around an organism. Osmosis involves the use of a partially permeable membrane, and the key word here is partially. It only allows certain molecules to pass through. Larger molecules are trapped and are not allowed to move. If we consider osmosis in practice, what happens if we place an animal cell in a high water potential solution, for example, pure water? Well, what happens is based enormously on the fact that an animal cell has no cell wall. So if you place an animal cell in pure water, water enters by osmosis, according to our definition, from an area of high water potential in the surrounding solution to an area of low water potential inside the animal cell. Across that partially permeable membrane, the water enters the animal cell and it keeps swelling and expanding until it can take no more and it bursts. And then we call this, if it occurs to red blood cells, hemolysis. If you were to place your red blood cell in a highly concentrated solution, so one with low water potential, this time water would leave your animal cell by osmosis, causing it to become shriveled and flaccid in appearance. It's now time to talk about the human skeleton. So first of all, we're answering the question, what is the purpose of the human skeleton? Well, it's to protect organs. So for example, your skull, your cranium, protects your brain, your eyes, your ears. Your rib cage protects your lungs, and your vertebral column protects your spinal cord. Getting into more detail now to do with the axial skeleton. The axial skeleton consists of the rib cage, the vertebrae, and the cranium. The functions of the axial skeleton is to protect your brain and spinal cord and to provide support. Moving on to the appendicular skeleton, what bones make up the appendicular skeleton? The scapula, so those are bones in the back of your shoulder, your clavicle, your pelvis, and your limbs. The function of the appendicular skeleton is to allow movement of the body. We're going into more detail now to do with the structure of the long bones. So first of all, the long bones are covered by hard, compact bone. This bone contains lots of calcium salts which make the bone hard and resistant to compression forces. The middle of the bone contains spongy bone. This has fewer calcium salts and contains spaces, and this means that that bone is less hard and less dense. The spaces found within that spongy bone contain bone marrow, which is responsible for producing blood cells, and it also stores fat. Not that I suggest anyone do this, but why does bone become soft when soaked in hydrochloric acid? It's because the hydrochloric acid dissolves those calcium salts. Without those calcium salts, the bone is much softer and less hard. What is a joint? A joint is a place where two bones meet. There are three classifications of joints: the freely movable joints, the partially movable joints, and the immovable joints. So the freely movable joints allow lots of movement. A good example here is the ball and socket joint. Partially movable joints only allow a small amount of movement. A good example here is the joints found in your vertebral column, so in your spine. Immovable fixed joints allow no movement at all, and the joints in your skull or your cranium are immovable. So the ball and socket joint, we've already said that it allows a lot of movement. It allows movement in all directions. Both your shoulder and your hip are examples of a ball and socket joint. They are both freely movable joints. A hinge joint allows movement in one plane only, such as your elbow. A gliding joint allows bones to glide past one another in any direction along the plane of a joint, such as the wrist and vertebrae. The general name given to a ball and socket, hinge, and sliding joint is a synovial joint. A synovial joint is a movable joint which contains synovial fluid. The synovial fluid is secreted by the synovial membrane. The purpose of the synovial fluid is to reduce friction between joints. It's oily in order to enable it to do this. It therefore acts as a lubricant. A capsule is a tough, fibrous material which surrounds a joint. Cartilage is strong material found at the end of a bone. The purpose of cartilage is to act as a shock absorber. What happens in someone with reduced cartilage or damaged cartilage is they have less of a shock absorber. It's going to become painful for them due to increased friction and the rubbing of bones. Moving on to ligaments. Ligaments are made up of tough fibers. The role of ligaments is to hold bones together within a joint. The various properties of ligaments include the fact that they're strong, which enables them to resist stretching. They're also elastic, which enables the bones to move without dislocating. You might have heard of the term antagonistic muscles, but you might not be entirely sure what it means. What it means is that as one muscle contracts, the other muscle relaxes. The triceps and biceps are examples of antagonistic muscles which help to move the arm. The biceps contract and the triceps relax in order to raise the forearm, and the biceps relax and the triceps contract in order to lower the arm. When the bicep relaxes, it becomes longer and thinner. When the triceps contracts, it becomes shorter and thicker, and this allows our forearms to become straightened. We now need to go into detail as to how voluntary muscles and bones bring about movement within a synovial joint. So we know that the muscles are attached to bone. The muscles work antagonistically as one muscle contracts, the other one relaxes, and this pulls on the bone, which brings about movement. What is a tendon now? Tendons attach muscle to bone. Unlike ligaments, tendons are inelastic, they don't stretch. Muscle contraction pulls on a tendon, causing the bone to move. A little more detail now about the vertebral column. So remember, here you'll find partially movable joints which allow little movement. The vertebrae are joined by discs of cartilage. Those cartilaginous discs provide shock absorbance. And altogether, the purpose of the vertebrae is to provide stability and support as opposed to free movement. You do not want your vertebral column moving. Healthy development of muscle and bones requires several dietary components. You need lots of protein in your diet for the growth and repair of your muscles. Vitamin D enables the uptake of calcium. Those calcium salts get absorbed at the gut and are used in the production of the hard material that bone's made up of. You'll often find that older women in particular suffer from a disease known as osteoporosis. Here the bone loses calcium salts and becomes more porous and less dense and more liable to being brittle and breaking. You find that women are more susceptible to osteoporosis due to the menopause when hormone levels decrease naturally. Unfortunately, there's no cure for osteoporosis, but a diet high in vitamin D and calcium can help. People with osteoporosis suffer from back pain and poor posture. Now I want to talk to you about coordination and the nervous system. So the function of neurons is to transmit electrical impulses. It's important you're aware of the three different neurons, the motor neuron, the sensory neuron, and the relay neuron. Be familiar with the shape of these neurons. Notice that motor neuron has that characteristic star-shaped cell body. The relay neuron sits between the sensory and motor neurons, so it is symmetrical at each end. Neurons have long axons in order to transmit the electrical impulse. Be aware of which direction that impulse passes along the neuron. The axon transmits electrical impulses away from the cell body. It has a very narrow region of cytoplasm surrounded by a cell membrane, and the axon itself is a single elongated nerve fibre. The cell body is where you'll find the nucleus located. Remember, that controls the activity of the cell. The cell body contains lots of endoplasmic reticulum and mitochondria needed to help produce neurotransmitters, which remember are transmitted across the gap between neurons. Also present on the neurons are things called dendrons. These are short extensions which come from the cell body. The dendrons divide into smaller branches known as dendrites, and the dendrons are responsible for transmitting the electrical impulse towards the cell body. The axon has a fatty layer surrounding it known as the myelin sheath. As with all fat, the fat acts as insulation and in this case helps to speed up the rate of the electrical impulse. If we zoom in on the sensory neuron transmits electrical impulses to either a relay neuron, a motor neuron, or the brain. The sensory neuron has a single axon which carries electrical impulses away from the cell body, and one dendron which transmits the electrical impulse to the cell body. The relay neuron sits between the sensory and motor neuron. It has short dendrons. The motor neuron transmits electrical impulses to an effector, which is either a muscle or gland. The muscle responds by contracting, the gland responds by secreting a hormone. Motor neurons have a long axon and short dendrites. The human nervous system consists of both the central nervous system, the CNS, this is made up of the brain and spinal cord, as well as the peripheral nervous system, the PNS, which is made up of effectively everything else. The role of the nervous system is in the control and regulation of bodily functions. It can both be under involuntary and voluntary control by the brain. Now we need to go into great depth with the structure and functions of the brain. So take a look at this diagram. Notice the two large cerebral hemispheres. You've got the cerebellum, the medulla, the pituitary gland, which is tightly linked with the hypothalamus. You've probably come across this when we've discussed various hormones such as ADH, LH, and FSH. The cerebral hemispheres are responsible for your conscious thought, your memory, your intelligence, language, and voluntary action. The cerebellum is responsible for muscle tone and posture. The midbrain is responsible for vision and hearing. The medulla controls your breathing rate and your heart rate. The pituitary gland is an endocrine gland which secretes hormones such as FSH, LH, and ADH. The hypothalamus is responsible for controlling blood water levels, known as osmoregulation, and your body temperature, which is known as thermoregulation. Responses can be split up into both reflex responses as well as a regular response. In a reflex response or a reflex arc, there is no conscious input from the brain. These are things which happen extremely fast, and the purpose of a reflex action is to protect us from harm, such as the time if you put your hand in a hot oven, you'll know that your hand withdraws itself without your need to register, oh, it's hot, oh, I don't want to burn my hand, oh, I must remember it. No. By that point, you'll have already removed your hand from the oven. So reflex actions are fast, they protect us from harm, and they don't involve conscious input from the brain. In order for our bodies to respond, we need something to respond to, and this is what's known as a stimulus. A stimulus is a change in the environment, so it could be a change in terms of whether you feel pain, whether there's heat, whether you're detecting something being hard or soft, whether you hear something, whether you see something. A receptor is a specialized type of cell which detects that change in environment. It detects the stimulus, so that could be your receptors in your eyes, your rods and your cones found in your retina, which are sensitive to light. Light is the stimulus in this case. There are lots of different receptors in your body which are sensitive to various different stimuli, such as light, pressure, temperature, taste, and pain. So if we think about what happens in a regular response, such as when our eyes detect light, say the rods detect low-level light, those are the receptors, then they're going to send an electrical impulse along the sensory neuron to your central nervous system, which is your brain, in this case. Then an electrical impulse is sent along your motor neuron to an effector. In this case, it will be a muscle in your eye, probably the ciliary muscle, which will either contract or relax based on how far away an object is from your eye. In a reflex action, the steps are slightly different. So let's talk about the reflex action involved when you step on a sharp pin. So the stimulus is the pain. The receptors are present in your foot. An electrical impulse is sent along the sensory neuron to a relay neuron located in the central nervous system. That impulse passes to a motor neuron and then finally an effector. In this case, that will be a muscle which responds by contracting in order to remove your foot from that painful pin. Do remember that between two neurons is a gap. This gap is called a synapse. It's important that you're aware that at a synapse, there's no electrical impulse. Instead, a neurotransmitter is released from the presynaptic membrane. This neurotransmitter diffuses across that synapse and binds to postsynaptic receptors. An example of a neurotransmitter is acetylcholine. Remember that electrical impulses can only pass in one direction across the synapse, and there's several reasons for that. Firstly, neurotransmitters are only released from the presynaptic membrane. Secondly, because diffusion is involved, the neurotransmitter only moves from an area of high concentration to an area of low concentration, so the high concentration will obviously be at the presynaptic membrane where that neurotransmitter is being released. And then thirdly, the postsynaptic membrane is the only place that contains receptors to receive that neurotransmitter. Now we'll turn our attention to hormones. So hormones are produced by a gland. They're secreted into the blood where they travel to a target organ. They're responsible for altering the behavior of that target organ, and we're going to talk a lot about various hormones right now. So we're going to start with the pituitary gland, which is located in the brain. The pituitary gland releases or secretes a large number of hormones, including LH, luteinizing hormone, which causes ovulation. That's release of an egg from an ovary. FSH, follicle stimulating hormone. This matures the egg in the ovary. ADH, antidiuretic hormone. Antidiuretic means effectively anti-peeing hormone, so it helps conserve water, and the mode of action of that ADH is in the collecting duct of the kidney. High levels of ADH increase blood water concentration. Now, adrenaline is responsible for preparing your body for fight or flight. So the classic examples is if you were confronted with a bear in the woods, would you stay to fight it or would you run away? I often think about my cat Lyra when I think about the effects of adrenaline because it's very clear to see it on her body. When you have high levels of adrenaline, your hairs stand up on end. This is to make you appear larger and more frightening. Your pupils dilate in order to help you see more clearly because that will allow more light to enter the eye. Your heart rate increases to deliver blood around your body faster. Your breathing rate increases in order to introduce more oxygen into your body. Remember, you'll need that oxygen for respiration because the respiration releases the energy you'll need to allow muscle contraction to help you fight or run away. So you'll find that blood is diverted away from your digestive system to your muscles in order to prepare yourself for that fight or flight. And that's why sometimes you feel what's known as butterflies in your stomach when you're a bit frightened because of the high levels of adrenaline coursing around your body. Glycogen stores are converted to glucose, again because glucose is needed in respiration, and I think I've already mentioned it, but you'll see increased levels of respiration throughout. Another hormone to be aware of is thyroxine, secreted by your thyroid gland in your neck. This controls your metabolic rate. Now we need to discuss the pancreas, specifically the islets of Langerhans, which are responsible for secreting the hormones insulin and glucagon. These both control our blood glucose levels. So after a meal, when your blood glucose levels increase, your body will want to reduce that. So insulin is secreted by the islets of Langerhans. The insulin causes blood glucose to be converted into insoluble glycogen, which is stored in the liver, therefore reducing blood sugar levels. When you haven't had much to eat or when you've exercised, what happens this time is the islets of Langerhans secrete the hormone glucagon. Glucagon causes the conversion of insoluble glycogen into soluble glucose, therefore increasing blood glucose levels. The gonads are sex organs now, so the ovaries secrete the hormone estrogen. Estrogen is responsible for secondary sexual characteristics in women. Really, this means the changes at puberty that occur, the onset of menstruation, that is blood loss, breast development, hips widening, pubic hair growth. Estrogen is also responsible for repairing the uterus lining. Now let's take the gonads in males. So what about the testes? Well, the testes secrete the hormone testosterone, responsible for male secondary sexual characteristics. Changes that occur at puberty for men include the voice deepening, pubic hair growth, sperm production. When you compare the hormonal system with the nervous system, the hormonal system is far slower, the effects are far more widespread. In terms of transmission type, you have chemical messengers carried in the blood versus the nervous system, which is much faster, a highly specific response, and we're obviously talking about electrical impulses carrying that information. Now let's discuss the eye. So remember, the eye is a receptor organ. It is sensitive to light and it's adapted in order to create a clearest image as possible. So if we go through the various parts of the eye, you've got your conjunctiva, which protects the eye. You've got the outer protective layer known as the sclera. You have your cornea, which is at the very front of your eye. This refracts light as it enters the eye. That light passes through the aqueous humor and then through the pupil. So the pupil's role is to control how much light enters the eye, and it's muscles in the iris that will control the size of the pupil. The muscles in the iris are both the circular and radial muscles. We'll talk a lot about that shortly, but like I've already said, the iris controls the size of the pupil. Moving backwards in the eye, we reach the lens. The lens carries out the fine focusing of the light. It refracts the light. Now the thickness of the lens alters how much that light is refracted, and the ciliary muscle and suspensory ligaments will alter the thickness of the lens. Then you have what's known as the vitreous humor. This is a pressurized liquid that helps maintain the shape of your eye. The all-important retina is located towards the back of the eye. It contains photoreceptors called rods and cones, which are sensitive to light. The rods are sensitive to low-level light. The cones are sensitive to color. You have a blind spot, which is where the optic nerve leaves the eye. No photoreceptors are found here. The fovea contains a high concentration of cones. You'll have the choroid, which is a black layer that stops light reflecting around the eye. Make sure you're clear that it's the lens which is responsible for focusing on objects which are different distances away from the eye. So you need to learn the steps as to what occurs if you're looking at an object close to and far away. So starting with scenario 1, looking at an object close to, here the ciliary muscles contract, meaning that the suspensory ligaments slacken and your lens is fat. This means that the light rays are refracted more strongly to produce a clear image on the retina. If you look at a far away object now, this time your ciliary muscles relax, meaning that your suspensory ligaments are pulled tight. This has the knock-on effect of making your lens thinner and light is refracted less strongly. Now we'll look at what happens in your eye when you're in different light intensities. So what happens if you enter a very bright room? Well, you'll want less light to enter the eye because you don't want damage being caused to the retina. So how do you make less light enter the eye? Well, you need to constrict or narrow your pupil. The way in which that occurs is that the circular muscles contract, the radial muscles relax, and your pupil constricts. If you enter a room with very dim light, this time you want your pupil to be nice and dilated, nice and wide to allow as much light as possible to enter the eye. So this time your radial muscles contract and your circular muscles relax. The major advantage of having two eyes is that it cancels out the blind spot. Remember, the blind spot is the place where the optic nerve leaves the eye. Having two eyes gives you a wider field of vision and allows stereoscopic vision. That's the ability to distinguish depth of field. A person may suffer from eye afflictions. Examples of this include long-sightedness, which is an inability to focus on nearby objects, short-sightedness, which is what I have, which is an inability to focus on objects far away. Cataracts may also occur, which is a cloudiness of a lens, as well as an astigmatism, which is when your eyeball is a slightly misshapen shape. So talking a little bit more about long-sightedness, remember that's an inability to look at objects close to. The problem here is that your rays converge behind the retina as opposed to producing a perfect image on the retina. So the reason for this is that your eyeball could be too short, or the lens of your eye is too flat. So the way to fix this is by using convex lenses. These are fat lenses, as they will allow that light to be bent more strongly. What about if you're short-sighted like me? Well, in this case, your eyeball might be too long, or my lens is too thick, meaning that the rays converge in front of the retina. In order to fix this, I need concave lenses, as this means that the light rays are refracted less strongly. More detail now on astigmatism. So you're going to get blurred vision, and that's due to your cornea being an uneven or irregular shape. You can use corrective lenses in glasses or contact lenses to correct this. As I've already mentioned, cataracts are caused by a cloudy lens. This is due to a buildup of protein. It's possible to have surgery to replace the cloudy lens. Damage sometimes occurs to the cornea, and it's possible to have a cornea transplant. The ear detects sound and is an organ responsible for helping us maintain our balance. There are 3 parts to the ear: the outer ear, the middle ear, and the inner ear. The outer ear consists of the pinna, so it's this portion of your ear here. Then you have the middle ear, which consists of the ear bones, the ear ossicles, the malleus, stapes, and incus, and then the inner ear contains the cochlea and the semicircular canals. Focusing on the outer ear then, the pinna, the role of the pinna is to collect sound waves and focus them onto the eardrum. The small bones which make up the middle ear, the incus, stapes, and malleus, their role is to amplify the sound waves. Don't forget about the eustachian tube. The eustachian tube connects the middle ear with the throat. It helps to equalize pressure either side of the eardrum, and it allows the eardrum to transmit vibrations. So now we're going to summarize how the ear actually hears things. So we need to start by discussing the fact that that pinna collects the sound waves, transfers the sound waves to the middle ear to the eardrum. The eardrum vibrates, and then the role of the middle ear, the ossicles, is to amplify the sound waves, to amplify those vibrations. The vibrations pass to the inner ear, specifically the cochlea, which contains hair cells which are sensitive to those vibrations. The hair cells in the cochlea send electrical impulses to the brain via the auditory nerve. So that's a summary as to how the ear hears sounds, but how does it maintain balance? Well, the important thing with balance is the semicircular canals. Crucially, the semicircular canals contain a fluid. As the head moves, the fluid in the semicircular canals move. This transmits information to the brain, specifically the cerebellum, which has an important role to play in maintaining our balance. It's obviously important that we take care of our ears. If we're exposed to a very large sound, like an explosion, it can cause damage to the ear bones, the ear ossicles, incus, malleus, and stapes. It can even cause the eardrum to burst, and this means that the sound waves can't be amplified properly. Prolonged exposure to a loud noise may lead to tinnitus, which is a ringing of the ear. It can even cause long-term hearing loss, and this is due to damage to those hair cells in the cochlea, meaning that no longer can electrical impulses be sent along the auditory nerve to the brain. It's now time to turn our attention to the drug topic. So starting with the definition of a drug is a chemical which has an effect on the body. So don't get confused, drugs aren't necessarily always bad things. I know the press will tell us that they are, but a drug is any chemical that has an effect on our body, such as paracetamol, which is a very valuable painkiller. It helps to reduce the temperature caused by a fever, but you must be careful not to take too much because an overdose may lead to liver damage and even death. What are the short-term effects of alcohol, which remember is a legal drug? Well, alcohol is actually a depressant. It slows the nervous system and reduces your reaction time. It can cause slurred speech, blurred vision, an inability to maintain your balance. So you'll notice that people who are drunk often stumble. And if you take too much in a short space of time, it may lead to a coma and even death. The long-term effects of alcohol now, it may cause huge damage to your liver, which is known as cirrhosis. It could cause gene mutation, which leads to cancer. It may cause brain damage, and it will affect your inability to detoxify substances due to that damage to the liver. If you do a deep dive into the effects of alcohol on the liver and the functioning of the liver, remember that the liver is an important organ for both detoxifying substances, but also in our digestion. Because the liver produces bile, any damage caused to the liver will affect its ability to produce bile. That means that fats can no longer be emulsified, which affects the working of the enzyme lipase. It also means that the pH can't be increased within the small intestine, and that's important to make sure that the enzymes released into the small intestine aren't denatured. So damage to the liver has very far-reaching effects. What about cannabis now? Cannabis is an illegal drug. It's a psychoactive drug. It causes feelings of relaxation, but with prolonged and long-term use, you can end up with hallucinations and feelings of disorientation. Cannabis is often smoked with tobacco, which may lead to damage to the lungs. Moving on to very nasty hardcore drugs such as heroin, a Class A drug. This is obviously an illegal drug. It is a painkiller. It's a narcotic, which means it gives feelings of intense pleasure. It's a depressant like alcohol, which means it slows the nervous system. The problem with taking heroin is that you build up a tolerance to it, which means that you need to take more and more heroin in order to experience the same feelings of pleasure. It's highly addictive, and there are some very nasty side effects associated with withdrawal from this drug, including diarrhea and vomiting. People often inject heroin, and the problem is people often use dirty or infected needles, so that causes the spread of HIV and hepatitis. Cocaine, another illegal drug, another psychoactive drug, causes intense feelings of pleasure. It also causes mental alertness, paranoia, and irritability. It increases your breathing and heart rate, so it's very, very bad for your heart, and it can raise your body temperature. It's highly addictive. Mental illness and disorders now. So we're going to consider schizophrenia and depression to begin with. So starting with schizophrenia, causes of schizophrenia can include genes, so hereditary, overuse of drugs, high levels of dopamine, and even stress. Symptoms of a person suffering from schizophrenia include hallucinations and paranoia, as well as anxiety and confusion. Treatments include cognitive behavioral therapy, CBT, and antipsychotic drugs. Now, depression has no one single cause, but a combination of factors may increase the chance of depression occurring. So it could be a traumatic event, bad childhood experiences, overuse of drugs, and poor physical health. There are lots of symptoms associated with depression, but it does include potentially suicidal thoughts, lack of energy and low self-esteem. Treatment options include antidepressant drugs and cognitive behavioral therapy again. Alzheimer's disease. Now, the cause of Alzheimer's is the buildup of proteins such as B-amyloid in the synapses in the brain. Lots of symptoms associated with Alzheimer's, including poor memory, lots of repetitive behavior, deterioration in social behaviour, loss of intellectual capacity, loss of voluntary muscle control. It's actually very difficult to treat Alzheimer's. You'll often find that people with Alzheimer's get treated with enzyme inhibitors, drugs which increase the release of neurotransmitters such as acetylcholine, and in general, activities which stimulate thinking skills. Let's understand why the buildup of proteins in the synapses in our brain affects us in this way. Well, remember, at the synapses, acetylcholine is released. It's a neurotransmitter which helps the progression of nerve impulses along the secondary neuron. When that B-amyloid blocks the synapse, it means that no new nerve impulses can be generated, which eventually leads to death of neurons. Why do enzyme inhibitors act as a treatment option for people with Alzheimer's? Well, enzymes are used to break down the acetylcholine at the end of a synapse. With an enzyme inhibitor, it means that you'll have higher levels of acetylcholine, meaning more electrical impulse transmission. Vascular dementia now, as the name suggests, it affects blood vessels supplying the brain. Here, the blood vessels become diseased and damaged, meaning less oxygen reaches the brain. The brain cells die. People with vascular dementia suffer from memory loss and difficulties with thinking and reasoning. Treatment options include drugs which reduce blood pressure and blood cholesterol, and general thinking activities. Parkinson's disease is caused by reduced dopamine levels. It occurs mostly in men and later in life. Because there's less dopamine, it means fewer nerve impulses are generated. Symptoms include tremors, a bent-over posture, stiffness, and difficulty moving. Treatments include providing drugs which mimic dopamine, enzyme inhibitors which prevent the breakdown of dopamine, deep brain stimulation, DBS, to reduce tremors, and more generally physiotherapy. Linking back to an earlier part of the spec, stem cell therapy is a potential treatment option for people with Parkinson's. Stem cells could be used to produce new brain cells that have been damaged in Parkinson's disease. So why is a balanced diet required for the healthy development of bone and muscle? Well, first of all, you need vitamin D because that allows the uptake of the all-important calcium ions also consumed in the diet, which will help strengthen your teeth and bones. You'll also need diets rich in protein as they're needed for muscle growth and repair. Make sure you're aware of the differences in the type of diets people require based on their activity, their lifestyle, etc. So people with more sedentary jobs will obviously require less food, so less carbohydrates, less protein, less fat, compared with people with very physical occupations. You often find that when girls reach puberty that they suffer from anemia, and that's due to a lack of iron brought on through menstruation, which, remember, is the blood loss associated with a period. Pregnant women will obviously need more of everything because they need to support the growing fetus in terms of its muscle growth, bone development, production of blood, so obviously we'll need more iron, more calcium, more protein, etc. in order to support that growing fetus. Defining digestion now. Digestion is the breakdown of large insoluble food molecules into small soluble ones, which can then be absorbed through the wall of the small intestine. There are two types of digestion. The first one is mechanical, the second one is chemical. So mechanical really means just breaking down that food into smaller pieces. There's no change in the chemical structure of the food. So if you think about it, the first place that mechanical digestion takes place is in your mouth. Your teeth chew, breaking up that food, increasing its surface area. A second place mechanical digestion takes place is in your stomach, where the stomach muscles contracting help to break down that food further, and hydrochloric acid in the stomach also assists with this. Chemical digestion really means the altering in the structure of the food. It means making that food more soluble, and it's enzymes which are responsible for this. So in the mouth, we've already discussed the mechanical digestion by the teeth, but also the enzyme amylase. This will be responsible for chemical digestion, the digesting of starch into maltose. Food then passes down the esophagus, so the bolus, the ball of food, passes down the esophagus with the aid of peristalsis, which is the contraction of muscles. Food reaches the stomach. I've already mentioned what the stomach does. Remember that hydrochloric acid also lowers the pH, meaning that any bacteria ingested will also be destroyed. Protease is an enzyme secreted by the stomach. It helps break down proteins into amino acids. Further peristalsis occurs in the small intestine. Remember that fiber is essential in your diet because it provides bulk to your food, it provides roughage, it aids the process of peristalsis, therefore helping prevent constipation. Moving on to bile. So remember, bile is produced by the liver, stored in the gallbladder, and released into the small intestine. The role of bile is twofold. Number one, it emulsifies fats, increasing their surface area, meaning that the enzyme lipase can work more effectively. Number two, it neutralizes the pH of food, meaning that enzymes released into the small intestine aren't denatured. We've already mentioned the enzymes amylase and protease. Remember the enzyme maltase. This breaks down maltose into glucose. You've got the enzyme lipase. This breaks down lipids into fatty acids and glycerol. The pancreas secretes all three enzymes into the small intestine. Now, the small intestine's role is to absorb those soluble food molecules. It's adapted in order to increase the rate at which absorption takes place. The microvilli and villi increase the surface area, meaning that absorption occurs more quickly. You've got the presence of a lacteal that aids fat absorption. You've got a good blood supply, lots of blood capillaries. Thin walls provide a short diffusion distance. The role of the large intestine is to reabsorb water. By this point, you'll have reabsorbed all your soluble food molecules, leaving you with just the indigestible foodstuff. This makes up the bulk of feces, which are stored in the rectum and released into the anus. Let's discuss some key definitions now. So ingestion, effectively eating, that's the intake of food. You've got egestion, which you mustn't confuse with excretion. Now, egestion is the removal of feces from the anus, so in effect pooping. Excretion is the removal of metabolic waste. Assimilation is the buildup of large molecules from small molecules. Digestion I've already mentioned. Absorption I've mentioned in passing. It's the movement of soluble food molecules across the lining of the small intestine into the blood. Now we'll turn our attention to the teeth. So, lots of different types of teeth. These ones at the front are known as your incisors. Then you have your canines slightly further back. You'll notice in dogs and cats that canines are very well developed. They're what gives you the fanged look. Then you've got your premolars and your molars. Your incisors cut and bite food. Your canines hold and cut food. Your premolars and your molars with that grinding surface are used to crush and grind food. If we think back to our childhood, so children have fewer teeth compared with adults. Their teeth are smaller. And remember, children lose their teeth around the age of 8. Adult teeth are permanent. You need a lot of information here about the structure of teeth. So starting with the outer casing, that's the enamel. It's the hardest surface found in the human body. It covers the tooth and provides a tough surface for biting and chewing. The root anchors the tooth to the gum. The dentine sits beneath the enamel. It's a bone-like substance which contains cytoplasm running from the pulp cavity, which supplies the tooth with nerve and blood vessels. The pulp is the hollow center part of the tooth, also containing nerves and blood vessels. Cement is the name of the substance which covers the root of the tooth. And then finally, blood vessels supply oxygen and general nutrients. Now we're answering the question, how does tooth decay occur? You'll notice if you have a diet high in sugar, you're more likely to get tooth decay, and that's because your tooth gets covered in the sugar. There are bacteria on your teeth which feed on that sugar. They use it as their food source. The bacteria aerobically respire. They convert the sugar to lactic acid, and the lactic acid degrades the tooth. Why are molars more susceptible to tooth decay? Simply because they're made up of ridges, so food gets stuck. They have a larger surface area. More bacteria collect here. Plaque is when bacteria form a coating on the teeth, and if it's allowed to harden, it forms what's known as tartar. In order to prevent tooth decay, obviously it's important to have diets low in sugar. You want to brush your teeth at least twice a day. You want to visit the dentist regularly, employ good dental hygiene, and actually tap water contains fluoride, which helps to harden the tooth enamel. The BMI is used to quantify whether someone is overweight or not. Obviously, it doesn't work for every single person, and that's because people who work out and have more muscle will register potentially in the obese category. But in essence, it's an equation. BMI, body mass index, is calculated by doing the mass in kilograms divided by height in meters squared. If you have a BMI less than 18.5, you're classified as underweight. From 18.5 to 24.9, you're healthy weight. 25 to 29.9, you're overweight. 30 and above, you are obese. If you have a BMI of over 40, you're morbidly obese, but remember, as I said, it doesn't count for everyone because of differences in people's lifestyle. People may become obese due to eating too much, not enough physical activity, or just a general predisposition to gaining weight. There are lots of health risks associated with being obese, including type 2 diabetes, coronary heart disease, and being a victim of strokes. Lots of factors contribute to coronary heart disease, including, as we've said, being overweight, being obese, smoking, a sedentary lifestyle, hereditary, so a genetic predisposition, drinking lots of alcohol, stress. Now, coronary arteries supply your heart with blood, and if you have diets too high in fat, what happens is a fatty deposit builds up in the lining of the coronary artery. It's known as a plaque. This narrows the artery, meaning that less oxygenated blood reaches heart muscle cells. With less oxygen means less aerobic respiration, more anaerobic respiration, which produces lactate. This lowers the pH, which denatures enzymes, ultimately leading to the death of heart cells. We've touched on diabetes, but now to talk about it in greater depth. There's two types, type 1 and type 2. Type 1, early onset, it has a genetic cause, and it's due to the pancreas not producing insulin, meaning that glucose cannot be converted to insoluble glycogen and be stored in the liver. The way we treat this is through the use of insulin injections. Type 2 tends to be from being overweight, having poor diets. This time, despite the fact that your pancreas releases insulin, your liver fails to respond to it. So careful control of diets and lots of exercise tends to be the treatment option here. Symptoms of diabetes include poor wound healing, loss of weight, which occurs rapidly, glucose in your urine, and frequent urination. The food hygiene topic now. So obviously food needs to be cooked, stored and preserved appropriately. So preservation methods, and I will talk about this in greater detail later on, these are used to stop microorganisms reproducing within the food. Cooking obviously kills any microorganisms which have contaminated the food. Storing is essential in order to make sure that microorganisms don't contaminate the food. How should food be stored in order to prevent contamination? Well, it's important that raw and cooked food is kept separately, so that's why they say in the fridge that raw food should be underneath cooked food. You don't want raw food leaking all over cooked food. You need to make sure that if you freeze food and then thaw it, that you eat it immediately. You don't want to keep reheating the same food. Now we're answering the question, how is yogurt made? So first of all, we need to pasteurize the milk that is used to make the yogurt. That means heating the milk to around 90 degrees Celsius. The purpose of pasteurization is to kill any bacteria present in the milk. We then need to cool that milk down to around 35 degrees and add the all-important Lactobacillus bulgaricus bacteria. This is the bacteria which converts the lactose sugar into lactic acid, giving yogurt that characteristic sour taste. We need to lower the temperature because high temperatures will kill that bacteria. And it's through the anaerobic respiration of that bacteria that the lactic acid is made. Now we're going to go into great depth about different food preservation methods. So I've already touched on pasteurization, so that's heating the food to around 90 odd degrees. That will kill any bacteria present. Rapid cooling will also prevent bacterial spores developing. Good examples of foods which are pasteurized include milk, yogurt and juice, beer and ice cream. Salting has historically been used, and this involves covering that meat or fish in salt. Any bacteria present lose water by osmosis due to that lower water potential caused by the salt, and that bacteria therefore die. Pickling, again, another popular historical method. This involves covering items of food in vinegar. The low pH denatures enzymes found in the bacteria. Fish, onions and some vegetables may all be pickled. I do know that onions are vegetables, by the way, before anyone comments. Canning involves placing food into a can, being heated to a high temperature. The can is sealed. The can is heated to over 100 degrees Celsius. This will kill the bacteria and spores. Fruits and vegetables, meat and fish can all be canned. Ultra-high heat treatment. This involves blowing steam through the food at super high temperatures of 160 degrees Celsius, killing all the bacteria and their spores. Milk is a great example of a food which undergoes UHT, and that's why you'll see on the side of some milk cartons UHT milk, but it does taste slightly different to fresh milk. Freezing now, so that means lowering the temperature to around minus 15 degrees Celsius. Although this won't kill the bacteria, it will absolutely slow their rate of reproduction. Drying methods now, hot air is blown through the food. Drying removes water from the food, meaning the bacteria can't use it and absorb it. Cereals and some fruits are dried. In some countries, irradiation is allowed, which involves food being exposed to gamma radiation, but like I said, only some countries allow this. Gamma radiation is a type of ionizing radiation. It will kill the bacteria and its spores. This can be used for vegetables, fish and shellfish. Respiration now. So aerobic respiration involves the use of oxygen. The word equation is oxygen plus glucose goes to carbon dioxide plus water plus the release of energy. Remember that energy is always released, it's never produced. Make sure you can write the symbol equation as well. So sixes are important here with the balancing. You want 6O2 plus C6H12O6, which is the formula of glucose, goes to 6CO2 plus 6H2O. Although energy is released, ATP is made. This is adenosine triphosphate. ATP is made from adenosine diphosphate, ADP plus an extra phosphate. And it's this ATP which is used to transfer the energy. Energy is released from ATP by its conversion back to ADP and the phosphate. We say that the ATP has been hydrolyzed. What is this energy released by respiration used to do? Well, it's used to help us contract muscles, move substances by active transport, carry out assimilation, which is building up large molecules from small molecules. It's also used in cell division as well as the manufacture of more organelles found within our cells. We've already said that we need oxygen for aerobic respiration. How does oxygen enter our body? Well, it enters through an inhalation, that's a breath in. The oxygen actually enters our blood in our lungs. Remember that general atmospheric air contains around 20% oxygen. It's mostly made up of nitrogen with some argon gas, carbon dioxide and water vapor. If we compare air that we breathe in, so that's inhaled air, compared with air that we breathe out, exhaled air, it makes sense that we absorb oxygen into our bodies. So therefore, exhaled air will contain less oxygen. Because carbon dioxide is produced by respiration, exhaled air will contain more carbon dioxide and more water vapor. So make sure that it makes sense to you. If you look at the equation for aerobic respiration, exhaled air must therefore contain more CO2, more water and less oxygen. Hydrogen carbonate indicator can be used to demonstrate how much carbon dioxide is present. So atmospheric air contains around 0.04% carbon dioxide. This turns hydrogen carbonate indicator orange. So if you see that that indicator is orange, you can say that there's around 0.04% CO2. High levels of CO2 turn the indicator yellow. Low levels of CO2 turn the indicator a red-purple color. Anaerobic respiration can occur if there's insufficient or a lack of oxygen. Here the glucose is incompletely broken down and it actually releases less energy and produces less ATP compared with aerobic respiration. Anaerobic respiration is carried out by yeast cells as well as our muscle cells if we experience intense periods of exercise. When yeast anaerobically respires, it takes that glucose and converts it into ethanol and carbon dioxide. This process is actually used in the bread making industry to help bread dough rise. The ethanol produced is used in the alcoholic drinks industry, so to make beer and wines. When our muscles anaerobically respire, this time the glucose is converted to lactate. Anaerobic respiration can cause muscle fatigue over time, and that's due to what's known as an oxygen debt. It means that when you stop exercising, your breathing rate stays high, and the reason for that is because you need to introduce more oxygen into our body in order to break down the lactate produced by anaerobic respiration. The respiratory system now, we'll start with its anatomy. So your windpipe, its scientific name is your trachea, which runs down here. Then it branches, quite similar to the branches of a tree really, into two bronchi. Smaller branches follow on, called bronchioles, and then it ends in air sacs, known as alveoli. You have a thick layer of muscle that sits towards the bottom of your thorax. That's called your diaphragm, which is able to contract and relax. Your rib cage provides a bony cage over the top of your lungs to help protect them. You've also got intercostal muscles, which are muscles which sit between your ribs, and they'll play an important role when you take a breath in and when you take a breath out. So I've already mentioned that the alveoli are air spaces. This is effectively where oxygen goes from your air in your lungs into your blood and carbon dioxide leaves your blood. There are small vessels known as capillaries which kind of cover your alveoli. They provide a very good blood supply in order to allow that gas exchange to take place. So what are the adaptations of the alveoli for gas exchange? Well, I've already said there's a large surface area, and that's due to their grape-like structure. They've got thin walls, so that provides a short diffusion distance. They're moist, which allows gases to dissolve, and they have good ventilation, which maintains a steep concentration gradient, and that means that diffusion of gases occurs much more quickly. Focusing in on the trachea, now the trachea is supported by rings of cartilage. They help to support the trachea, stop it from collapsing, and they also enable your neck to bend. The pleural membranes now, these are thin, moist membranes. They act to separate the inside of the thorax from the lungs, basically to stop your lungs sticking to the thorax wall. The gap between the two membranes is known as the pleural cavity, which is filled with pleural fluid, and this fluid acts as lubrication, again stopping the lungs from sticking to the wall of the thorax, because remember, those lungs need to inflate and deflate to allow gas exchange to occur. So focusing in now on ventilation, that's the movement of air into and out of the lungs. What about an inhalation, a breath in? The following changes occur. First of all, your diaphragm contracts, it flattens, your external intercostal muscles contract, and together these cause an increase in the volume of your thorax. Because the volume's increased, by definition the pressure must decrease, and that's really in comparison to atmospheric air, and as a result air is drawn into the lungs. What about an exhalation now, a breath out? The opposite occurs. This time your diaphragm relaxes, it becomes dome-shaped, your external intercostal muscles relax. Together, these reduce the volume within your thorax, meaning that the pressure this time increases compared with atmospheric pressure. Therefore, air is forced out of your lungs. If you consider the difference in composition between inhaled and exhaled air, clearly inhaled air will contain more oxygen compared with exhaled air because the oxygen's been used by cells in respiration. Exhaled air contains more carbon dioxide and more water vapor compared with inhaled air, and that's because these two substances are products of respiration carried out by our cells. So these substances get transported to the lungs so that they can be breathed out. Now we're getting into the nitty-gritty of some kind of intense lung terms, such as lung capacity. This is the maximum volume of air that can be contained within our lungs. Vital capacity is the maximum volume of air that can be forcefully exhaled from our lungs in one breath. Residual volume, this is the volume of air left in our lungs after a forced exhalation. And tidal volume, this is the volume of air breathed in one normal breath. A spirometer is a piece of equipment or apparatus that can be used to measure the volume of air breathed in and breathed out. There's a mouthpiece that you breathe in and out of, which is attached to an air chamber which moves up and down. It's attached to a pen which draws a trace on a piece of paper. Why does our breathing rate increase during exercise? I'm sure you're aware if you've gone on a run, you'll start breathing far faster, far more deeply. The reason for that is because your muscle cells in particular need to respire more. They need to release more energy to allow your muscles to contract. Respiration, remember, produces carbon dioxide. This increase in carbon dioxide is detected by the brain, specifically the medulla oblongata, as well as receptors in the arteries. Electrical impulses are sent to the respiratory center in the medulla, which causes an increase in breathing rate in order to remove that extra carbon dioxide from the blood. There are obviously lots of benefits associated with exercise on the cardiovascular system. It includes increased numbers of red blood cells, increase in the size and strength of the heart, decrease in resting heart rate, increased muscle mass and bone density, increased number of alveoli, and a decrease in resting blood pressure. Let's now discuss 2 ways in which the heart rate may be increased. I'm sure you're already aware during exercise. The reason for that, as I've already said, is because there's more carbon dioxide being released into the blood. That gets detected by receptors in the aorta and the carotid artery, sends electrical impulses to the medulla oblongata in the brain, and the accelerator nerve is then important for increasing the heart rate. The hormone adrenaline, which is released when we exercise, is also responsible for increasing the heart rate. One of the most straightforward methods we can use to measure our heart rate is by taking our pulse rate. Remember, you can feel that pretty much in your wrist and in your neck, although you might want to attach yourself to a monitor in order to get a more accurate representation of your pulse rate. The simplest way to see the effect of exercise on the heart rate is basically to take your pulse rate at rest for 1 minute, then do some exercise, and then measure your pulse rate directly afterwards for a further minute. Remember, the advantage of an increased heart rate during exercise is because the heart is responsible for pumping blood around your body. That blood contains the all-important glucose and oxygen needed by cells to carry out the respiration required to release energy and to enable our muscles to contract. You find that the fitter the person, the more they exercise, the lower their pulse rate, their heart rate tends to be. Now we'll talk about the effect of smoking on the body. So smoking is obviously really bad for you. There's lots of reasons for this. First of all, cigarettes contain tar. Tar is a carcinogen. This is a chemical which causes cancer. So people who smoke for a very long time may see increased likelihood of getting lung cancer. The other problem with smoking is that it paralyzes those cilia which line your trachea. Remember, these are cells which waft backwards and forwards, removing bacteria-laden mucus from our trachea so that it can be destroyed by the hydrochloric acid in our stomach. When our cilia is paralyzed, that bacteria stays in our trachea. Therefore, you see an increase in infection and an increased incidence of diseases like bronchitis. Long-term smoking may also cause the disease emphysema. Now, emphysema damages your alveolar walls, therefore effectively reducing their surface area, meaning that less gas exchange takes place. So a person really struggles to breathe out when they have emphysema, which is a chronic disease that can't really be treated. You'll often see people using oxygen tanks if they have emphysema. Cigarettes contain nicotine. It's an addictive drug. Nicotine actually increases the viscosity of the blood. It makes it thicker, meaning that you have an increased chance of high blood pressure and the coronary heart disease associated with that. Cigarette smoke contains carbon monoxide. Carbon monoxide, remember, combines irreversibly with the hemoglobin in red blood cells. This prevents the transport of oxygen, so it is highly toxic. Focusing in on the blood. So what are the components of the blood? Well, you have both white blood cells and red blood cells. Remember, white blood cells consist of both lymphocytes and phagocytes. Red blood cells, their official name is erythrocytes. Remember, they're needed to transport oxygen. You've also got a jelly-like substance known as plasma and small fragments of cells called platelets. And we'll talk lots about their various roles a bit later on in this video. The blood is responsible for transporting oxygen from the lungs to respiring cells, carbon dioxide from respiring cells to the lungs, various nutrients such as glucose and amino acids, hormones, urea, which is a breakdown product of proteins from the liver to the kidneys where it's excreted. Kind of repeating ourselves a little bit, but just talking about the role of the plasma now. So the plasma, the jelly-like substance, transports hormones from where they're made, glands, to the organs that they act upon, their target organs, nutrients such as glucose from the small intestine to cells, urea from the liver to the kidneys, carbon dioxide from respiring cells to the lungs. So what I've just mentioned there is specifically the role of plasma, whereas before I was talking more generally about the role of blood as a whole. Something you might be less familiar with is tissue fluid. This is fluid found between capillaries and the cells. It effectively bathes the cells. How is tissue fluid formed? Well, the artery end of the capillary has a higher pressure compared with the venous, the vein end. The capillary walls are leaky. Water and small molecules are forced out of the blood due to this pressure difference. Larger molecules such as proteins remain in the blood. If we consider the difference in the composition between tissue fluid and plasma, you'll find that tissue fluid contains no large proteins, no red blood cells, unlike plasma, which may contain both of these things. How does water leave the tissue fluid? Well, water moves by osmosis from an area of high water potential in the tissue fluid to an area of low water potential in the capillaries across a partially permeable membrane. What is the lymphatic system? Well, it's a system of vessels containing the substance lymph. Lymph contains water, fatty acids, and lymphocytes. Areas of the lymphatic system where you find nodes are known as lymph nodes. These contain a high concentration of lymphocytes. Remember that the lacteals found the small intestine play an important role in forming lymph. They aid fat absorption. Focusing in on red blood cells, so as I've already said, red blood cells are responsible for transporting oxygen around the body. That oxygen binds to a pigment known as hemoglobin present on the red blood cells. The red blood cells have lots of adaptations to increase how much oxygen they transport. They have a characteristic biconcave disc shape. This increases their surface area to volume ratio, maximizing the amount of oxygen that can be transported. They have no nucleus. This provides more space for hemoglobin. They're flexible, which enables them to squeeze through blood capillaries, and they have a thin membrane which provides a short diffusion distance. Have you ever wondered why athletes like to train at high altitude? It's because of the following. At high altitude, you find there's less oxygen present. To compensate, more red blood cells are produced by our body. More red blood cells means that there's more hemoglobin, meaning that more oxygen can be transported by our blood. More oxygen is great for athletes because it means that more respiration can take place, more energy released, and more muscle contraction. Now we're answering the question, how are blood groups determined? This is due to the presence of various antigens. Remember, these are small proteins which stick out, in this case from the outside of the red blood cells. There are two types of antigen, A and B. This allows us to have 4 different blood groups, A, B, AB, and O. As you would expect, someone with blood group A has A antigens on their red blood cell surface, B has B antigens, AB has both A and B antigens, O has neither A or B antigens. There are no antigens sticking out of the red blood cells of group O. Now remember that small proteins known as antibodies will attach onto these antigens, so let's consider those various blood groups in turn to work out what type of antibodies will respond to those antigens. So for blood group A, they have antigen A sticking out of their red blood cells. Therefore, antibodies found in blood group A people will be anti-B antibodies. For blood group B people with B antigens poking out of their red blood cells, this time they'll have anti-A antibodies present in their blood. This means that they'll attack any red blood cells with A antigens sticking out from them. AB this time, blood group AB will have AB antigens poking out. Based on the fact they have both A and B antigens, it means that they have neither anti-A or anti-B antibodies. And then what about blood group O? Well, we know this time there are no antigens sticking out, so this time this person's antibodies will react to both A and B. So they'll have anti-A and anti-B antibodies. So what does that actually mean in terms of blood donation? Sometimes people need a blood transfusion for a variety of reasons. Let's consider which blood group would count as the universal donor. So the universal donor means that this blood can be given to anyone regardless of their blood group. Well, that universal donor blood group must be O. The reason for that is because they have no antigens on their red blood cells, meaning that when their blood is given to other people, there's nothing for the antibodies in that other person's blood to respond to. Hence why blood group O is known as the universal donor. Now let's consider which blood group is known as the universal recipient. It means that this person can receive blood from anyone. Well, in this case, it's going to be the person with blood group AB. The reason for that is because their red blood cells already have both A and B antigens poking out from them, meaning it doesn't matter where the blood comes from, they won't have antibodies which attack because they don't attack their own cells. So blood group AB is known as the universal recipient. If the wrong blood is given to a person, a process called agglutination occurs, which is when the antibodies attach to the antigens on the red blood cells, causing the red blood cells to clump together. This is very dangerous due to the blockage of blood vessels that may occur. Unfortunately, blood can never be frozen because it causes the red blood cells to burst, and therefore the blood is effectively useless. The white blood cells form the body's defense against pathogens. Remember, pathogens are microorganisms which cause disease. They include things like bacteria, fungi, protists, and viruses. So there are two white blood cells. The first type is the phagocyte, the second type is the lymphocyte. Phagocytes have a characteristic lobe nucleus. Phagocytes carry out phagocytosis. What this means is that they engulf the pathogen. They do this through that lobe nucleus. It means that they can engulf that pathogen into their cytoplasm, whereupon digestive enzymes are released to destroy the pathogen. The lymphocytes, these have a large single lobe nucleus. This is because they don't engulf pathogens. Instead, they recognize small proteins on the pathogen surface known as antigens. The lymphocytes secrete small proteins known as antibodies, which bind to the antigen. Now, the lymphocytes, when they secrete these antibodies, first of all, the antibodies can help kill the pathogen, the bacteria. How do they do that? They can help cause the bacterial cells to clump together, making them more likely to be spotted by phagocytes so that they can then be engulfed. They also produce antitoxins, which counteract any toxins produced by the pathogen. And another method by which they may destroy the pathogen is by causing them to burst open. What about those small fragments of cells known as platelets? So, to simplify their role, we would say that platelets clot the blood at the site of a wound. So as soon as you cut yourself, you don't bleed indefinitely. And the reason for that is because your platelets create a scab at the site of a wound. But the process is slightly more complicated than that. So what happens when you have a wound is that the platelets accumulate at the site of a wound. The enzyme thrombokinase is released from the platelets. This enzyme causes the conversion of prothrombin to thrombin, which in turn causes the conversion of soluble fibrinogen into the insoluble version, fibrin. Fibrin is the substrate for the enzyme factor 13. The fibrin meshwork formed traps red blood cells, meaning that a scab is formed. And the real importance of all of this is to ensure that you don't have excess blood loss and that pathogen entry is prevented. If you're doing human biology, it is important that you're aware of the role of thrombokinase, prothrombin and thrombin, as well as fibrinogen and fibrin. Some people have a lack of thrombin, and this really affects their ability to carry out blood clotting in the following way. Lack of thrombin means that fibrinogen is no longer converted to fibrin. There's no longer any substrate for that enzyme, and the fibrin can no longer form a meshwork to trap the red blood cells needed to form a clot. Now we turn our attention to discuss various blood vessels, starting with the arteries. You need to be able to describe their structure and function. So if we start with their structure, we say that arteries have a narrow lumen. This means that the hole that the blood flows through is very narrow. That's the lumen. Because of that, blood flows at high pressure, and to stop the blood vessels bursting, they need thick muscle and elastic fibre walls to withstand that pressure. Arteries carry blood away from the heart. Arteries tend to be oxygenated. The exception here is the pulmonary artery, which takes blood from the heart back to the lungs. That is deoxygenated. Veins have a much wider lumen. Because of this, blood flow is much slower at much lower pressure, and therefore they have thinner walls made of muscle and elastic fibres. Because that blood flows at such low pressure, sometimes it has a tendency to back up, and that's why it's a valve which ensures that blood flows in the correct direction. Veins tend to be deoxygenated. They contain no oxygen. The exception here is the pulmonary vein, which transports oxygenated blood from the lungs to the heart, specifically the left atrium. The capillaries now, these are much smaller than arteries and veins. They're only 1 cell thick. They have thin walls to provide a short diffusion distance. Their walls have small pores that allows the movement of substances. Capillaries have a very large surface area to volume ratio. Pulmonary means relating to the lungs. Renal means relating to the kidneys. Hepatic means relating to the liver. Use these words to help you label the various blood vessels around the body. If the main artery coming from the heart is known as the aorta, it feeds out of the left ventricle. Any vessel coming off the aorta therefore must be an artery. So, the name of the vessel supplying the liver is the hepatic artery. That supplies oxygenated blood to the liver. The vessel supplying oxygenated blood to the kidneys is the renal artery. Now if we turn our attention to blood vessels flowing away from organs, because these vessels will join the vena cava, they must be veins. Let's take The vessel leaving the liver, it must be the hepatic vein. The vessel leaving the kidneys must be the renal vein. Don't forget that the hepatic portal vein exists between the intestines and the liver. This will contain the highest concentration of glucose because it's coming from our gut. Do think about various other components of blood coming into and out of organs. It makes sense that blood going to the lungs must have the highest concentration of carbon dioxide, the lowest concentration of oxygen. Blood leaving the kidneys, remember the kidneys are an organ of excretion, therefore blood in the renal vein must be lowest in urea as that's been excreted in our urine. The heart is a muscular organ which helps to pump blood around the body. It's very important that it can do that because of what I've said previously. The blood transports a large number of very useful substances, the most useful of which is oxygen needed by cellular respiration. So in terms of the anatomy of the heart, it has 4 chambers, 2 atrium and 2 ventricles. Just be very careful when it comes to labelling the heart. The left and right sides are flipped compared with what you'd probably think, and that's because if you picked up the diagram and popped it into your chest, that's how the heart would actually appear. So notice that you have the left atrium, the left ventricle, the right atrium and the right ventricle. The separation between the left and right side of the heart, it's called the septum. A septum simply means a wall of muscle, such as your septum in your nose here. It keeps the left and right hand sides of the heart totally separate, and the reason for that is it stops the mixing of oxygenated and deoxygenated blood. The left side of the heart contains oxygenated blood. That means blood containing oxygen. The right hand side contains deoxygenated blood. That means blood containing no oxygen. So that's your broad layout of the heart. Next up, you have vessels which supply the heart. Any vessel that comes into the heart is known as a vein. Any vessel which leaves the heart is known as an artery. Arteries carry blood away from the heart. Try and remember that those 2 A's come together. Right, let's get into the nitty-gritty of the heart then. So we're going to start with the left atrium. I've already told you that the left side of the heart is oxygenated. So where must that blood have come from? Well, oxygenation, gas exchange, occurs at the lungs. So blood supplied to the left atrium comes from the lungs, and the name of this vessel is the pulmonary vein. The left atrium contracts, forcing blood into the left ventricle. The left ventricle contracts, forcing blood into the main artery known as the aorta. This artery goes off and splits into smaller arteries supplying various organs and cells with oxygenated blood. The cells use up that oxygen in their respiration, so we say that the blood becomes deoxygenated. It obviously needs to return to the lungs in order to become oxygenated, but in order to do that, it needs to go to the heart first of all. It does this by travelling in the vena cava vein to the right atrium. The right atrium contracts, forcing blood into the right ventricle. The right ventricle contracts, forcing blood into the pulmonary artery. This blood is then returned to the lungs, where it can pick up more oxygen. There are various valves to prevent the backflow of blood in the heart. Between the walls of the left atrium and the left ventricle, you'll find the bicuspid valves, the left atrioventricular valves. They're both names for the same thing. And then in the walls between the right atrium and the right ventricle, you'll find the tricuspid valve, the right atrioventricular valves. These prevent backflow of blood back into the atrium. At the base of the pulmonary artery in the aorta, you have what's known as semilunar valves. These valves prevent backflow of blood into the ventricles. Remember, it's important that the left and the right hand side of the hearts are separate in order to prevent oxygenated and deoxygenated blood from mixing, as this would result in lower rates of respiration. We say that the human heart is a double circulatory system. The reason for this is that blood enters the heart twice for every 1 circulation around the body. If you look more closely at the anatomy of the heart, you'll notice that the wall of the left ventricle is thicker than the wall of the right ventricle. Why is this? Well, that thickness means that there's more muscle. And the reason why the left ventricle needs more muscle is it needs to generate a higher pressure to force blood much further around the body. Whereas remember, the right ventricle is simply pumping to the lungs. How is the pulse rate created? Remember I said the pulse rate is the same as the heart rate. Well, blood leaves the heart in short bursts, causing the stretching to the wall of the aorta. When the ventricle relaxes, stretches of the aorta recoil, which increases the pressure within the aorta. Next up, the coronary arteries. Now, the heart is the hardest working muscle in the body. It contracts myogenically all by itself throughout your lifetime. Now, the heart needs a huge amount of oxygen in order to carry out those contractions, and that's the role of the coronary arteries, which snake over the outside of the heart. The coronary arteries supply the heart with its own oxygen requirements. Now, what happens if you lead a fairly unhealthy lifestyle? If you eat too much saturated fat, potentially smoking, it can be caused by stress. It can even be hereditary, so caused by genetic factors. But what may occur is that fatty deposits may build up in the lining of the coronary arteries. This narrows the lumen of the artery, meaning that less oxygenated blood reaches the heart muscle cells. As a result of this, aerobic respiration can no longer occur, more anaerobic respiration occurs, leading to the buildup of lactic acid, and fundamentally the heart muscle cells die. There are various treatment options for coronary heart disease, including a stent. Here you have a small metal meshwork which is placed in the occluded artery. There's a balloon inside the meshwork which gets inflated, forcing that meshwork to open up that artery. Then the balloon is removed and you've still got that meshwork in place holding open the coronary artery. This obviously requires intense surgery. It's quite a dangerous operation. It is a long-term solution, but there's always the risk of pathogen entry as soon as you make an incision in the body. If complete heart failure has occurred, then someone will require a heart transplant. The problem with this is they obviously come from other humans. You need a heart of exactly the right size, and there is always a shortage of heart transplants. In the meantime, an artificial transplant can be used. This is made up of plastic, fibers and metal alloys. An artificial heart is not a long-term solution. Ideally, you would require a human transplant, but remember there's always issues with rejection. When tissues don't match, blood groups don't match, the person will require immunosuppressant drugs for the rest of their life in order to stop the heart from being rejected. Remember that rejection occurs because there are antigens, small proteins on the side of the new person's heart. These get recognized by the person whose body it's going into's lymphocytes, and antibodies are produced in order to attack the antigens on that new heart. Hence, it's the lymphocytes which need suppressing. Another option could be a heart bypass, where effectively a new vessel is fashioned, redirecting blood away from the blockage in the coronary artery. Statins are a drug used to treat coronary heart disease. They work by lowering blood cholesterol, which is also a contributing factor to coronary heart disease. Statins work by inhibiting an enzyme involved in the production of cholesterol. Sometimes they ask you to compare the relative advantages and disadvantages of statins versus heart transplant. Use your common sense here. The advantage of statins is obviously that they're cheaper, they're more readily available, they're easier to take, there's no major operation. Problems include the fact that someone might actually forget to take them, and there could be possible side effects. Another potential treatment option includes plant stenol esters. Plant stenol esters have a very similar structure to cholesterol. They help to lower blood cholesterol in our bodies, therefore reducing the risk of coronary heart disease. There are several different heart diseases. Obviously, we've mentioned coronary heart disease. What about angina? Well, angina is pain in the heart caused by coronary heart disease. Beta blockers are a type of drug which may be used to treat angina. Beta blockers work by binding to adrenaline receptors, meaning that adrenaline can no longer bind. Remember, one effect of adrenaline is to increase the heart rate. So if the adrenaline binding sites have been bound to, it means that adrenaline can no longer take effect. The heart rate can no longer be increased. So beta blockers, in essence, lower the heart rate. Moving on to blood pressure now. Two types of blood pressure, systolic blood pressure and diastolic blood pressure. Systolic is the highest blood pressure reached. The systolic pressure is caused when the left ventricle contracts, and the normal range for systolic heart pressure is between 100 and 140 millimeters of mercury. The diastolic pressure is the lowest pressure caused when the ventricles relax, and your usual range for diastolic blood pressure is between 60 and 90 millimeters of mercury. Hypertension is the term used to describe chronic high blood pressure. Lots of reasons for hypertension. It could include stress, obesity, smoking. Again, it could be hereditary, caused by genetic factors, drinking alcohol, high levels of salt in the diet. Treatment options for hypertension include beta blockers again, which remember lower the heart rate. ACE inhibitors, remember ACE stands for angiotensin converting enzyme inhibitors, as well as consuming a healthy diet and taking plenty of exercise. So ACE inhibitors, now angiotensin converting enzymes are responsible for producing angiotensin. Angiotensin narrows blood vessels, therefore increasing blood pressure. So therefore, if you think about it, ACE inhibitors prevent angiotensin being made, therefore meaning that less blood vessel narrowing occurs, meaning that blood pressure is effectively reduced. The dreaded monoclonal antibodies, please don't worry about this. I'll take you through this topic step by step. We need to first of all understand what various definitions mean. So first of all, what is a clone? It's a genetically identical cell or individual. So what are monoclonal antibodies? Well, antibodies, remember, a type of protein. The antibody binds to the antigen. The key thing about monoclonal antibodies is that they're produced from a single clone of cells, meaning that all the antibodies made are genetically identical. They're all clones of each other. So with monoclonal antibodies, we're really trying to think of a way that we can produce large numbers of antibodies. That's the real purpose of monoclonal antibodies. And we know that antibodies are produced by lymphocytes, but the problem with lymphocytes is that they reproduce incredibly slowly. So what scientists thought about next was, well, how do we get them to produce large numbers of antibodies? What type of cell replicates really quickly? And they thought, hang on, a cancer cell, a tumour cell. So what happens if we combine a tumour cell with a lymphocyte? Maybe we'll be able to generate large numbers of genetically identical antibodies, and that's exactly what they did. They combined a tumour cell with a lymphocyte to produce a hybridoma. Remember, hybrid is a little bit of one thing and another, such as the Toyota Prius. Remember, that's a hybrid using both petrol and electricity in order to power itself. So to go into the nitty-gritty as to how monoclonal antibodies are made, we first of all need to get a mouse. The mouse is injected with a specific antigen, and before long the lymphocytes of the mouse start producing antibodies specific to the antigen which has been injected. It's actually the mouse spleen cells which are used to produce those antibodies. The spleen cells are fused with tumour cells to produce a hybridoma, and it's the hybridoma cells which are responsible for making large numbers of monoclonal antibodies. So what are the uses of monoclonal antibodies? Why have we gone to all this effort to combine lymphocytes and tumour cells? Well, they can be used in diagnosis, such as in pregnancy tests. Pregnancy tests contain monoclonal antibodies. The monoclonal antibody binds to the HCG hormone present in a pregnant woman's urine, and by binding to a dye, you see this as a positive test result in a pregnancy test window. Monoclonal antibodies may also be used for testing in laboratories to detect the presence of a particular pathogen, hormone levels or chemicals within the blood. Monoclonal antibodies can even be used to treat cancer. The antibodies can be attached to radioactive substances which can be used to kill cancer cells. Going a little more into detail as to how monoclonal antibodies can be used to identify pathogens, one end of the antibody is attached to a fluorescent dye or marker. The other end attaches to the specific antigens on a pathogen. The moment that that binding occurs, the fluorescent dye shows up and you can view the pathogen under a microscope. Homeostasis means the maintenance of a steady internal environment. Such internal environments include body temperature, glucose concentration of the blood, as well as water levels of the blood. It's important that our body temperature remains fairly constant because if it rises too high, enzymes may be denatured, killing cells. If it's too low, then enzyme-controlled reactions occur too slowly. The skin is super important in thermoregulation. That means the control of our body temperature. But the skin has several other roles. First of all, it acts as a barrier preventing pathogen entry. It contains melanin, which absorbs harmful UV rays from the sun, helping to prevent skin cancer. It's also an organ of excretion. Remember, the skin excretes sweat. But how does the skin help us control our temperature? How does it carry out thermoregulation? Let's think about when we're too cold. When we're too cold, our hairs on our skin stand up on end, trapping a layer of insulating air close to the skin, meaning that we're kept warmer in this way. Other mechanisms involved include shivering when our muscles contract, releasing heat. Vasoconstriction occurs in the arterioles in our skin. Remember, vasoconstriction means the narrowing of these arterioles, meaning that less blood flows close to the skin, less heat is radiated. Sorry about the seagulls. What about when we're too hot? This time you'll see vasodilation occur. This is when the arterioles dilate, more blood flows close to our skin, more heat is radiated. Our hairs on our skin lay flat, less insulating air is trapped. Sweating occurs. As sweat evaporates, it helps to cool the body. The layer of the skin are as follows. You've got the epidermis, which is the top layer, the dermis, and then the subcutaneous fat, which sits underneath this layer. Thermoregulation is a good example of negative feedback. This means that when a change occurs to the environment, mechanisms are brought about within our body to oppose that change. So, for example, Using when we're too hot as an example, first of all we need to detect the change in temperature. Thermoreceptors detect that we're too hot. Nerve impulses are sent to the hypothalamus. The hypothalamus sends nerve impulses to the skin, increasing sweating, causing our hairs to lay flat, causing vasodilation to the arterioles supplying our skin. As a result of all these processes, our body temperature decreases and our normal body temperature of 37 degrees Celsius is restored. And then the opposite occurs if we're too cold. But again, the hypothalamus is responsible for regulating all the processes that will help increase our body temperature. A second type of homeostatic mechanism you need to be aware of is known as osmoregulation. Remember, osmo means anything relating to water, so osmoregulation is all about controlling our blood water levels. The super important organ involved with osmoregulation is the kidney, but do remember that the kidney has two roles. It controls blood water levels, but it also excretes the substance urea. Remember, excretion is the removal of metabolic waste from our bodies. Substances which are excreted include sweat from the skin, carbon dioxide from the lungs, and then focusing in on the kidney, urea from the kidney, and that urea is excreted in urine. You have two kidneys. They're two fist-sized organs found in the lower portion of your back. They have various vessels which feed into and out of them. The blood vessel supplying oxygenated blood to the kidneys is known as the renal artery. The blood vessel removing deoxygenated blood from the kidneys is known as the renal vein. In terms of the layers of the kidney, the outermost layer is known as the cortex, the middle portion is known as the medulla, and then you have what's known as the pelvis. Now, the kidney contains millions of tiny structures known as the nephron, and it's important that you know the structure of the nephron inside out. The blood vessel which enters the nephron is known as the glomerulus. It's important that you're aware that the structure of the glomerulus is such that the blood vessel coming into it is wider than the blood vessel leaving, and that has important implications when we consider pressure. The first part of the nephron is known as the Bowman's capsule, and then you enter the first coil tube, which unfortunately you need to be able to describe its full name of proximal convoluted tubule. We move into the loop of Henle or loop of Henle. Then we move into the second coil tube, known as the distal convoluted tubule, before entering the collecting duct. That feeds into the ureter, which supplies the bladder with urine. Now, at the glomerulus Bowman's capsule boundary, a process known as ultrafiltration occurs. So ultrafiltration really means small molecules such as water, ions, urea and glucose are moved from the glomerulus into the Bowman's capsule under pressure. Now, that pressure comes from the fact that the blood vessel entering the glomerulus is wider than the one leaving, generating pressure to force those small molecules out of the blood. Do note that things like proteins are too large to enter the Bowman's capsule and they remain in the blood. And if there's any protein present in your urine, then that can indicate damage to either the glomerulus or the Bowman's capsule, because it means the pressure is so great that those proteins have entered the nephron when ordinarily they would stay in the blood. The next process you need to be aware of is known as selective reabsorption. So if you consider the contents of the nephron, it contains ions, urea, glucose, water. Obviously, it's important for our body that we retain as much glucose as possible. Otherwise, what was the point of us eating food containing glucose? The point of our digestive system trying to absorb that glucose through the wall of the small intestine is because we want that glucose in our blood. But at this point, it's entered the proximal convoluted tubule. So selective reabsorption is really taking back that glucose and some ions back into the blood. It occurs at the proximal convoluted tubule, and crucially it occurs by active transport, and that's because the glucose moves from an area of low concentration to an area of high concentration. Therefore, there's the requirement of energy from ATP. So what that means is that the urea is allowed to stay in the nephron, where it joins the ureter, is added to the urine, and at that point can be excreted from the body. So selective reabsorption and ultrafiltration are really important in the excretion of urea. What about the osmoregulatory role of the kidney? So we're going to take two scenarios, one when we've had too little to drink and one when we've had a lot to drink. So what happens when we have too little to drink? Well, obviously we want to retain as much water as possible in the blood, and the following steps occur in order to do this. So first of all, we have osmoreceptors present in the hypothalamus in our brain. These detect the low water levels of our blood. They send a signal to the pituitary gland to release more of the hormone ADH. ADH stands for antidiuretic hormone. The ADH travels in the blood to the nephron, and specifically it acts on the walls of the collecting duct, making them more permeable to water. This means that more water is reabsorbed into the blood, less water is available to make urine, so resulting urine is concentrated, yellow in colour, low in volume. But what about when we've had lots to drink? Well, we're going to just learn those same steps but use slightly different wording. This time our osmoreceptors in the hypothalamus detect high water levels in the blood. The pituitary gland therefore secretes less ADH. The ADH travels to the collecting duct in the nephron. It makes the walls of the collecting duct less permeable to water, meaning that less water is reabsorbed, meaning that resulting urine is high in volume and dilute and light in colour. Where does the substance urea actually come from? Well, our bodies are unable to store excess protein. That protein gets broken down into amino acids. The amine group is removed from the amino acids in a process known as deamination, and this actually takes place in the liver. Remember, if there's glucose present in your urine, that also indicates there's a malfunctioning of the kidney, because remember that glucose should all have been reabsorbed at the proximal convoluted tubule. If there's glucose present, then it means that something has happened at the proximal convoluted tubule. Now to discuss the control of blood glucose, another example of homeostasis. So after a meal, you're going to have lots of glucose in your body, and your body can't just store that glucose as it is, because glucose is highly soluble, meaning that it has a strong osmotic effect. It draws water effectively into and out of cells. So in this case, what happens when you've had lots of glucose in your diet? Your pancreas will secrete the hormone insulin. Insulin causes the conversion of soluble glucose into insoluble glycogen, and that glycogen is stored in the liver, therefore decreasing blood glucose levels. What happens if you haven't eaten in a while? This time, your pancreas secretes the hormone glucagon. The hormone glucagon causes insoluble glycogen to be converted to soluble glucose, and that glucose is released into the blood, therefore increasing blood glucose levels. This is yet another example of negative feedback. When a change occurs, processes are brought about which oppose that change that was made. So in the case of when you've eaten lots of sugar, processes occur to decrease our blood sugar levels, and then the opposite occurs if you haven't eaten any sugar. I've already outlined all the ways in which the kidneys are super important because of their roles in osmoregulation and the excretion of urea. But what happens when kidney failure occurs? What are the various treatment options available? So you need to know about two treatment options. Number one is dialysis, and number two is a kidney transplant. The way you can think of dialysis is that it's a machine used to clean the blood. And by cleaning the blood, I mean it's used to remove the urea, which, remember, if allowed to build up, becomes toxic. So how does it work? Well, the patient first of all needs to have their blood removed from their body. The blood passes through the machine, and there's a semi-permeable membrane with the dialysis fluid running alongside. The important thing you need to know is that the dialysis fluid contains the same concentration of glucose, ions and water as what's present in the blood, but the dialysis fluid contains no urea. So the person's blood flows past the membrane, and due to the fact that there's no urea in that dialysis fluid, urea leaves the person's blood by diffusion, moving from an area of high concentration to an area of low concentration. The blood is returned to the person's body, this time with no urea. It's been effectively cleaned. Because the concentration of glucose, ions and water remains constant in the dialysis fluid, it's the same level as that found within the person's blood, none of those things leave the person's blood, so that means that it gets returned when the blood passes back into the person's body. It's important that the glucose and ions don't leave, because after all, glucose especially is needed for respiration in order to release energy in cells. Anti-blood clotting chemicals are added to the blood as it enters the dialysis machine to prevent clots blocking the machine. It needs to be added at the start of the process rather than at the end, because after all, you need the usual clotting properties in the blood as it goes back into the person's body. Now, the thing about dialysis is it has to be carried out several times a week. It takes hours and hours, so it really does affect the person's quality of life. It does lose effectiveness over time, so it's not a super long-term solution, and a very careful control of diet is required. You can't be eating too much protein if you're going through dialysis. The reason for that is because, remember, through the process of deamination, proteins are broken down, the amine group is removed from the amino acid, urea is produced. You really want to limit the quantity of urea made, so that's why a carefully controlled diet is required. The other issue with dialysis is because it involves multiple puncturing of the skin, this is a potential source of infection. Pathogen entry may occur here. So again, not ideal. The second option for someone with kidney failure is a kidney transplant. So this is when a healthy kidney is transplanted into the person's body who's suffering with kidney failure. The advantage here is there's no need for dialysis. It's a much better quality of life. It remains effective throughout. However, it does require the use of immunosuppressant drugs in order to prevent our immune systems rejecting the new kidney. And there's always risks involved with operations, although there's less puncturing of skin. It's not something which occurs constantly, as is the case in dialysis. It's just one operation. Now, kidneys tend to be donated more often than other organs in the body. The reason for that is because people are born with 2 kidneys and they can function perfectly well with just 1 kidney, so it makes it more likely that a kidney will be donated. But as with all organ transplants, the list of donors is never as high as the number of people requiring a kidney transplant. Now to really unpick the role of the liver. So I've already mentioned the role of the liver in the digestive system is to produce the substance bile. Remember, bile emulsifies fats. That means it breaks down large fat droplets into smaller fat droplets, increasing the surface area, meaning that the enzyme lipase can work more efficiently. The second role of bile is to neutralize acid which has been added in the stomach, meaning that enzymes added in the small intestine aren't denatured. The liver also carries out deamination. This means removing the amine group from the amino acids. Remember, amino acids are produced from the breakdown of proteins. Excess protein cannot be stored by the body. So through a number of processes, that protein is broken down into amino acids, and then finally the substance urea is made, which, remember I said, is excreted in urine at the kidney. Another important role of the liver is to detoxify, particularly substances like alcohol. The liver also helps to regulate the quantity of glucose in the body. Remember I said that if you've had lots of glucose to eat, excess glucose can't be stored. It needs to be converted into glycogen through the action of the hormone insulin. That insoluble glycogen is stored in the liver, so that's another role of the liver, to store glycogen. Now, the liver has an unusually high rate of blood flow. The reason for that is because it receives blood from both the hepatic artery and the hepatic portal vein, which, remember, is coming from the small intestine. The liver is also a very hard working organ, needing a rich supply of blood due to its roles in detoxification and deamination. If people drink too much alcohol, several things can happen. Their liver will become damaged. It will struggle to detoxify substances like alcohol, can lead to scarring if there's lots and lots of drinking, and this is known as cirrhosis. There's an increased chance of gene mutation leading to cancer, and less bile will be produced by the liver. As a result, you won't be able to neutralize stomach acid so well, and your fat droplets won't be broken down into small fat droplets as effectively, meaning that the surface area to volume ratio of the fat droplet remains small, so therefore the enzyme lipase can't work as efficiently. So effectively you can't digest fats as well if your liver is damaged. Now we move on to reproduction. So first of all, we need to start with a sperm and an egg cell. The sperm cell is what we call the male gamete. It is the male sex cell. The egg cell is the female gamete, the female sex cell. Remember that sperm are made in the testes under the control of the hormone testosterone. Eggs are made in the ovaries. Several hormones are important here: follicle stimulating hormone, luteinizing hormone, estrogen and progesterone all play a role. The moment that egg is released from the ovary, it then travels along the fallopian tube where it waits to be fertilized. The sperm enter the female's body during copulation. The sperm are deposited in the vagina. The sperm swims through the cervix, up through the uterus, where it fuses with the egg in the fallopian tube. This is the process of fertilization. Now, do remember that sperm and egg cells are both haploid. They contain 1 set of chromosomes each. The reason for that is that when they fuse at fertilization, that the first cell formed, the zygote, contains a diploid set of chromosomes. It contains 2 sets of chromosomes. So ensuring that the sperm and the egg have half the number means that the diploid number is restored when that zygote is first formed. In order to form our embryo, the zygote must then divide. The type of cell division responsible for that is called mitosis. So we'll go from 2 cells to 4 cells to 8 cells, 16 cells, and so on. Meiosis is a different type of cell division. Meiosis is used to produce the sperm and the egg cells, so meiosis takes place in the ovary and the testes. Meiosis is essential to this process because it introduces variation. It ensures that every sperm and every egg is genetically different, and that's important when we consider the offspring produced. Survival of the fittest states that there will be variation within a population, and those organisms better adapted are more likely to survive. So meiosis is one way of introducing variation. The other way is through random fertilization. You do not know which egg is going to fuse with which sperm. And this is why sexual reproduction has an advantage over asexual reproduction because of the variation produced. It helps in a changing environment. With asexual reproduction, when there's only the need of one parent, all offspring produced are genetically identical to each other and their parent, their clones. Although this is a super fast reproduction method, if conditions do change, it does mean that it's likely that all the organisms will die because none are better adapted to a particular environment. Let's focus in on great detail as to the structure of the male and female reproductive systems. So starting with the male reproductive system, I've already mentioned you have the testes. These are responsible for sperm production and it's under the influence of the hormone testosterone. There are two testes and they're held in a sac of skin known as the scrotum. The purpose of the scrotum is to, in effect, hold those testes outside of the body. This helps to lower the temperature, and it's that lower temperature which is vital for healthy sperm development. When that sperm is released, it passes along the sperm duct and various fluids are added from the prostate gland as well as the seminal vesicles. These provide nutrients to help the sperm to swim. The sperm then passes down a tube called the urethra. This will help convey that sperm as well as urine out of the male body. So the urethra is a tube which passes along the entire length of the penis and it helps to convey sperm and urine out of the male body. The penis, as I've already said, it allows urine to exit the body. It also, in effect, places sperm within the vagina of the woman during copulation. And then lastly, the epididymis. This is responsible for storing sperm. Now we move on to the female reproductive system. So we'll start with the ovaries. The ovaries make the eggs. Remember, ovulation occurs, which is the release of an egg from the ovary, which is under the control of the hormone LH, luteinizing hormone. The eggs enter the fallopian tube. The other name for the fallopian tube is the oviduct. There are small cilia here. These are small hairs which help to move the egg down the fallopian tube where it sits and waits for any sperm. If copulation, sexual intercourse occurs while the egg is sitting in the fallopian tube, the person may become pregnant, the zygote, and then the embryo forms and the embryo implants in the wall of the uterus. So the uterus is where your fetus, your embryo, will grow for the 9 months, which is the gestation period for a human female. Gestation period just means how long she's pregnant for. The role of the cervix is to act as a boundary between the uterus and the vagina. It holds the growing fetus in place and forms the first part of the birth canal. The vagina is where sperm's deposited during sexual intercourse. It's also known as the birth canal because the baby will have to pass down the vagina in order to exit out of the body. Females also have a urethra, which is a tube used to convey urine out of the body. Now we'll talk a lot about the menstrual cycle now. So this is a cycle lasting approximately 28 days. It helps prepare the woman's body for a potential pregnancy. So we're going to be talking a lot here about hormones. So the first hormone you need to be aware of is FSH, follicle stimulating hormone. As the name suggests, it stimulates the follicles. These are the immature eggs found within the ovary. So in effect, FSH matures the egg. The next hormone that's important is known as LH, luteinizing hormone. This causes ovulation, the release of an egg from the ovary. In the meantime, you've got estrogen levels increasing. Remember, estrogen is produced by the ovary. Estrogen is responsible for thickening that uterus lining ready for implantation. Progesterone is produced by both the ovary and the placenta later on in pregnancy. This helps maintain the thickness of the uterus lining. LH and FSH are made, remember, in the pituitary gland, so their source is actually the brain. As well as its impact on the uterus lining, estrogen also has a role of inhibiting FSH and stimulating the release of LH. Progesterone has the role of inhibiting the release of FSH and LH, and that makes sense. If you've already got an embryo developing in the uterus, the last thing you want is more eggs maturing and being released from the ovary. Now, what happens if no sperm enters the vagina? The uterus lining is no longer needed. It doesn't need to be thick anymore, so effectively it breaks down. It is shed, and this is what we call menstruation, and it's the blood loss which is associated with a period. So menstruation just means blood loss. It is the shedding of the uterus lining. Now, the placenta is a hugely important organ. It develops while the fetus is developing inside the uterus. It's an organ supplying the fetus with everything it needs for growth. So it will supply oxygen needed for respiration, glucose again needed for respiration. It will supply amino acids needed to develop muscles within that fetus, vitamins again needed for bone development, so that will be vitamin D as well as the calcium mineral ion. The placenta is also important for removing waste products produced by the fetus such as carbon dioxide and urea, and the placenta is adapted in order to ensure the transfer of these materials happens as efficiently as possible. It has villi which increase the surface area, meaning that diffusion of substances can occur much more quickly. It also has a good blood supply, lots of blood capillaries, and thin walls provide a short diffusion distance. The amniotic fluid is found within the amniotic sac, and it bathes the growing fetus, effectively acting as a shock absorber, meaning that the fetus is less vulnerable to any physical impact. What are the stages of birth? Well, the amniotic sac tears, meaning that the amniotic fluid is released, and that's what we call the waters breaking. The cervix dilates, and that enables the fetus to start its movement down the vagina through the birth canal. The baby is born, and then there's the delivery of the afterbirth, which is actually the placenta leaving the woman's body, because after all, she doesn't need it anymore. The hormone oxytocin is incredibly important in childbirth. It helps the uterus walls to contract, helping to deliver that fetus. Obviously, there's lots of different ways to feed the baby. Various advantages with breastfeeding. First of all, breastfeeding is the total optimal food. It provides all the nutrients, the glucose, the amino acids in the exact right amounts for that growing baby. It also provides passive immunity. This means that antibodies are transferred from the mother to the baby, giving protection against pathogens. It's a good way for the mother and baby to bond, and it reduces the risk of diabetes and allergies later in life. Stages of growth in the human lifespan. So directly after birth, there's a huge increase in growth rate. This is known as the infant growth spurt. The growth rate decreases slowly up until the age of 4. Obviously, that infant's still growing, and that growth rate slowly decreases again up to the age of 10. And then as puberty hits, there's another huge growth spurt. This is known as the adolescent growth spurt. A person's growing is done by the time they reach their late teens. Organs also grow at different rates. You'll have organs like the kidneys and liver which keep up with the overall growth rates. Reproductive organ growth rate is very slow from the ages of 0 to around 13, and then as puberty hits, due to the release of those sex hormones, you'll see a huge increase in growth rate in the reproductive organs. Organs like the brain grow very quickly early on in childhood. I'm sure you'll notice that when babies are born, they have a very large head. The growth rate of the head decreases in proportion, whilst the length of the limbs increases in proportion. It's useful to measure growth rate as a proportion of the whole body in babies, and that's to account for differences in height. You can't just say for one person, oh, their legs aren't growing properly. You need to look at their growth of their legs as a whole compared with the rest of their body, as opposed to compared to the rest of the population, because people simply are taller and shorter based on variation. Percentile charts are used to check that a baby's growing healthily. Their weight is regularly compared to the percentile chart, and it helps doctors to work out if the baby's growing too much in mass or too little in mass. Various methods of birth control, so ways in which you can prevent yourself from becoming pregnant. So the simplest method is a barrier method, something like a condom or a femidom. In the case of a condom, a plastic sheath worn over the penis, it stops sperm reaching the egg. Other options include oral contraceptives, that means taking a pill. These pills often contain estrogen and progesterone, because remember, these hormones both inhibit FSH production. Without FSH, eggs can't mature. Hormone injections and implants in your arm work in a similar way. They release estrogen and progesterone, inhibiting FSH and preventing egg maturation. Then you've got intrauterine devices such as the coil. This is like a metal which is placed inside the uterus. It stops eggs implanting, meaning that no fetus can develop. The rhythm method simply means not having sexual intercourse when you're fertile. So when that egg's been released, that method is really unreliable, because everyone's menstrual cycles vary in length. People ovulate for different lengths of time, so it's not particularly recommended. For a more long-term approach, you can have a hysterectomy or a vasectomy. A vasectomy is when the sperm ducts are tied. A hysterectomy is when the oviducts are cut. Both of these stop sperm and eggs reaching various places. It's quite hard to reverse both the vasectomy and the hysterectomy. Remember, when considering which method to use, things like condoms and femidoms, these are good because they stop sexually transmitted diseases spreading, whereas oral contraceptives will have no effect on STDs. And then, of course, there's always abstinence, which means not having sex at all. Remember that with the exception of the hysterectomy and vasectomy, which tend to be 100% reliable, the other methods can't be guaranteed to avoid pregnancy. With things like oral contraceptive, it obviously requires the woman to constantly remember to take her pill. There can be various side effects such as gaining weight, mood swings, and even an increased risk of blood clots. But again, these are very unlikely side effects, so please don't stress. Some couples struggle to get pregnant and they might need some fertility treatment to help them. The most popular type of fertility treatment is IVF, which stands for in vitro fertilization. So what happens is the woman is given injections containing LH and FSH. The FSH causes her eggs to mature and the LH causes ovulation. So you get huge numbers of eggs developing. These eggs are harvested and placed in a petri dish where semen is added from the male partner. Fertilization may occur, meaning that embryos are formed, and then those embryos are placed into the uterus of the woman. Often several embryos are used. So obviously this is a great option for people struggling to get pregnant. It does have a relatively high success rate, but it is stressful. It's not guaranteed, and it does involve the woman in particular to receive lots of injections. The risk of multiple births has increased due to the implantation of several embryos, but several embryos do need to be placed inside the uterus to increase the chance of success. We now turn our attention to the hereditary topics, so genetics. We need to start by going through a whole list of various definitions, and it is important that you know the meaning of all these terms so you can apply these things to the questions when you're asked. So starting with an allele is a form of a gene, such as having blue eyes or brown eyes. They're both alleles for eye colour. They're given by letters, so blue eyes being a recessive trait will have a lowercase b denoting it. Brown eyes being a dominant trait will have a large uppercase B denoting it. A gene is a section of DNA which codes for a particular protein. The genotype is the alleles belonging to an organism. You'll have two alleles, so if you have brown eyes, for example, you could be uppercase B, uppercase B. Blue eyes would be lowercase b, but that is your genotype, whereas your phenotype is the physical appearance of a trait. So that is whether you have brown eyes or blue eyes. It is not the same as the genotype. Make sure you're clear on that, and we will look at some past paper questions to help you here. Homozygous means that you have two of the same alleles, such as two lowercase b's or two uppercase b's. Heterozygous means that you have two different alleles, such as a capital B and a lowercase b. A recessive trait, the alleles are only expressed in the homozygous state. That means you'd need lowercase b. As soon as that uppercase b is present, then the dominant trait would be expressed. Dominant is when only one copy of an allele is needed to express a particular trait. This is different to codominance, where both alleles are present in the phenotype. A good example here is the snapdragon plant. If you have a red snapdragon flower, a white snapdragon flower, rather than one colour being dominant over the other, what would happen is that both alleles would contribute to the phenotype, and in this case you'd see a pink flower. Moving on to haploid and diploid. Haploid means containing only one set of chromosomes. In humans this is 23 chromosomes. Diploid means containing two sets of chromosomes. In humans this is 46. Monohybrid inheritance means that one gene is responsible for a particular phenotype. Polygenic inheritance means that many genes are responsible for a phenotype, and in actuality that does tend to be the case for most traits. In most cases, many genes contribute to a particular phenotype. So let's take a simple genetic cross. A heterozygous brown-eyed mother and homozygous blue-eyed father want a baby. What is the probability that their baby will have blue eyes? So we're going to use the following layout in order to make sure we answer this properly. Please don't rush this. You want to write mother, father, phenotype, genotype, gametes. So this is the preparation you do in order to make sure that you get your Punnett square nice and accurate. So set yourself up like this. You should have plenty of space in the exam to do this. Now look at the clues given in the question. We're going to use B to represent eye colour. So we know that we have a heterozygous brown-eyed mother. The fact that it's heterozygous means that the genotype must contain one uppercase B and one lowercase b. We know that the father is homozygous and blue-eyed. Blue, remember, is a recessive trait. Homozygous means that those alleles are the same. So there's the father's genotype. We're now ready to write down the answers in our table. So the phenotype of the mother is that she has brown eyes, because remember, phenotype is the physical appearance. The father has blue eyes. Now list the genotype. We know the mother, I've already worked it out, is capital B, lowercase b. Father is lowercase b, lowercase b. The gametes, this means the mother's sex cells, so this is the egg cells. Her egg cells will either have a big B allele or a lowercase b allele. The father's sperm are all going to be that lowercase allele. This means you're now ready to set up your Punnett square. So it should look something like this. Just list those alleles again. It's up to you if you circle them. Give yourself plenty of space. And the Punnett square will enable you to work out the possible likelihood of that baby having blue eyes. So just list the various genotypes. And you should give yourself space to list the phenotypes underneath. So this baby here will be brown-eyed. This one will be blue-eyed, brown-eyed, blue eyes. So in terms of answering the question, our probability is that there's a 0.5 chance of the baby having blue eyes. Although percentages aren't technically probability, you'd also get away in biology with writing 50% chance or 1 in 2 chance. But it's important that you do this because these questions tend to be worth around 4 marks, and that will enable you to get the marks for linking the various phenotypes and genotypes at the end. So I do implore that you spend the time writing this out properly. Let's take an example now involving cystic fibrosis. So we have a carrier mother and a carrier father of cystic fibrosis. Cystic fibrosis is a nasty disease caused by the recessive allele. I'm going to call that C. So in order to have cystic fibrosis, you need 2 lowercase c's. So the carrier mother automatically tells me that her genotype must look like this, the same for the father. It's because they contain that lowercase allele, which is responsible for causing cystic fibrosis, but because it's a recessive disease, they've got this healthy allele, which luckily for the carriers means they don't suffer with any of the effects. But if we actually have a look at the cross this time, we're going to use the exact same layout as before: mother, father, phenotype, genotype, gametes. Like I said, it works every time. So you can write healthy or carrier here, it's up to you. I've already worked out the genotypes. So half of those egg cells will be healthy, half will contain that lowercase c, same for the sperm. And then if we look at the Punnett square, that's a healthy child. Here is a carrier, still healthy though. It's that final child here which will have cystic fibrosis. We can say that these ones here are carriers and that this one's entirely healthy. So it's 25% chance or 0.25 chance of having cystic fibrosis. I've already said that a diploid number of chromosomes means 23 pairs of chromosomes. One pair is actually known as the sex chromosomes, and this determines whether someone is male or female. Males have an XY sex chromosome combination, females have an XX chromosome combination, and it's possible to do a genetic cross to show this. Let's now look at sex inheritance. So we know that the mother is going to be female, the father is male. I've already said that the sex chromosomes in females is XX, in males it's XY. So that means all of the female's eggs will contain an X. Half of the male sperm will be female, the other half will be male because they'll contain a Y. And again, you're ready to get going. So this baby here will be female, this baby here will be female, this baby here will be male, this baby here will be male, hence why there is a 50-50 chance of having a girl versus a boy. Pedigree analysis is used to show how a particular genetic condition is passed on from generation to generation. They are in effect family trees, so you will have the grandparent level, the parent level, the grandchild level, and you'll be able to see a male and a female and the children they produce, and then using the key you'll understand whether they have a particular condition or not. The great thing about pedigree analysis or pedigree tree is it allows you to work out whether a particular condition is a recessive or dominant condition. So for example, if two healthy parents, and you'll be able to use the key to work out if they're healthy or not, have a child with the condition, it tells you that the condition is recessive and that both parents must have been carriers. That's the only thing that can be true if they're both healthy but their child has the condition. Equally, you'll be able to work out based on the number of people suffering from a particular disorder whether it's a dominant condition or not based on how many people have it in that pedigree tree. And we'll look at an example now to really take you through step by step so you can work out the sort of thinking you'll need to unpick these questions. Familial hypercholesterolemia, FH, is an inherited condition caused by a dominant allele. People with the condition have high levels of cholesterol in their blood, and this increases the risk of dying from blocked arteries. The diagram shows the pattern of inheritance in several generations of a family with FH. So as I said, here's the all-important key down here that will tell you that all the squares are males, all the circles are females. If it's shaded in, then they must have the disease. If it's blank, then they are healthy. So the first thing you want to do is work out what genotypes would be required to cause FH. We know it's a dominant condition, which means that either this combination or this combination will cause the disease FH. The only way you can be healthy is to have the recessive genotype, which looks like this. It's very important that you get to grips with what the information is telling you. So if we look at part A, person A is heterozygous for FH. Use this information to complete the table. So we've been told that this person has two different alleles. We can immediately label all of the unaffected healthy people's genotypes because I've already said they must be lowercase h, lowercase h. So we're being asked how many people are homozygous recessive. Homozygous means they have two of the same allele. Recessive tells me that they're both lowercase. So then it's simply a matter of just counting up all of the homozygous recessive people. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. What about homozygous dominant? Homozygous, two of the same allele. Dominant tells me that they're both capital H. So now we need to work this out. This daughter here has the disease. We know that she'll have only got a lowercase h from her mum. What about this son over here? Same setup. He has the disease, so he must be uppercase, but he's got that lowercase h from his mum, and you just keep working it down. Look, it's the same combination here, so I can immediately write that allele combination, that genotype, then working down to person N. They'll have inherited the lowercase h from their mum, the uppercase h from their dad in order to have the disease. The same is true for all three offspring. So actually, if you sit back and look, how many people have this genotype? Well, the answer here is 0. Person G and person H have 3 children who all have FH. What is the probability of G and H having 3 children who all have FH? So G and H are here. Let's do a super quick Punnett square to work out the answer to this. So these two people here will be diseased, so 0.5 chance of having the disease because they have this genotype. So in order to work out the chance of them all having FH, because it's probability in maths, remember if it's person 1 and person 2 and person 3, you need to multiply that probability 3 times to get 0.125 as your answer. Some disorders are sex-linked, which means that the allele for the particular trait is located on the sex chromosome, so that would be the XX chromosomes in females, the XY chromosomes in males. And you often find that with sex-linked characteristics, they're more common in one sex compared with another. For example, red-green colour blindness is way more common in men compared with women. Most sex-linked diseases are actually located on the X chromosome. The reason for that is because the Y chromosome is much shorter and therefore has comparatively fewer genes located on it. Just think about the length of an X and a Y in the alphabet and you'll see that the X is longer. X-linked dominant traits are more common in females, and there's quite an obvious reason for this. It's because they have 2 X chromosomes, which means they have 2 alleles. So either allele may be dominant and cause disease, so it doesn't matter if it's homozygous or heterozygous. If it's dominant, it will cause the disease in the female. Whereas X-linked recessive traits are much more common in males. The reason for that is they have only 1 X chromosome, and therefore there's no potential for masking by another allele because quite simply it doesn't exist on the Y chromosome. If they have that copy, even if it's that lowercase allele, the recessive trait, the fact that they don't have a dominant allele on another X chromosome to mask that means that they'll suffer from the disease. So we can make the following conclusions: that for sex-linked diseases, it's only possible for females to be carriers. Males always inherit their X-linked allele from their mother, and females cannot inherit their X-linked allele from an unaffected father. Red-green colour blindness is a recessive sex-linked disorder. The allele for colour blindness is located on the X chromosome. A heterozygous female has a child with a normal-sighted male. What are the possible genotypes and phenotypes of the offspring? So we're going to go female, male. This setup works for all questions. Phenotype, genotype, gametes. So we've been told that red-green colour blindness is recessive. So in order to have the disease for the female, they need to have this allele combination, this genotype. So we know based on the fact that they have this uppercase that they must have normal vision. We've been told that the male is normal-sighted, so I can write that here. The genotype they've already given to us. What are the gametes? So we'll just separate those nice and carefully. Now we can draw a Punnett square. So now we're ready to list our possible outcomes for these offspring. Okay, so in terms of, we're going to have a female here with normal vision. Here's a male with normal vision because they have that uppercase C. Here's a female, normal vision. Technically this one's a carrier. But then this one down here is a male with colour blindness because they have that lowercase c responsible for the red-green colour blindness. So in terms of outcomes, it's 0.25 chance of being a male that's colourblind. We're now going to move on to discuss haemophilia. Now, haemophilia is a blood clotting disorder. It's caused by a recessive allele. That recessive allele is located on the X chromosome only, so it is a sex-linked disease because it's located on the X chromosome. So if we consider various genotypes, there's a female that will be healthy. Here's a female that again will be healthier but a carrier due to the presence of that allele there, which remember is the recessive allele responsible for haemophilia. Here's a male that will be healthy. And here's a male that will have the disease. And that's due to this lowercase h here. And remember, we said that the allele for haemophilia is only located on the X chromosome, so that's how I can infer all these things. Now let's look at a Punnett square cross. Haemophilia is a blood clotting disorder caused by a recessive allele found only on the X chromosome. A man needs to inherit only one allele to have the disease. What is the probability of a male haemophiliac and a female carrier having a haemophilic child? So, ready to go again. So male has the disease, female is healthy. We can infer that from the wording of the question. The genotype, so here is the sex chromosomes for the man. We know that he's a haemophiliac, which means he must have that lowercase h in order to have the disease. Remember that allele will only be located on the X chromosome. The female, we've been told, is a carrier. There's her female sex chromosomes. For her to be a carrier, she'll need that genotype. So we've done all the hard work. Let's do the Punnett square now. So this is a female healthy carrier. This is a male who's healthy. This is a female with the disease. And this is a male with the disease. Huntington's chorea is a dominant genetic condition that results in the nervous system not working correctly. It is not a sex-linked condition. Diagram 1 shows a family pedigree. Some members of the family have the condition. Remember to use that key so we can see straight away. Here we have a healthy male, a diseased female, healthy male, female, healthy male, diseased female, healthy male, diseased male. Evaluate the evidence in the pedigree that supports the statement that this is not a sex-linked condition. It's because if you consider the individuals which are affected, 3 of them are female, 2 of them are male, so we can make the point first of all, it's not a sex-linked condition because it appears in both males and females in similar numbers. Evaluate the evidence in the pedigree to support the statement that this is a dominant condition. So dominant means that you would expect to see lots of sufferers, and indeed you see people suffering in every generation. You could even work out some of the genotypes. So we'll use H to represent. That woman there must be large H, lowercase h. The reason I know that is because her partner here is definitely lowercase, because he doesn't have the disease. And if you look at their children, we can see that this male here doesn't have the disease, this female does, this male doesn't, this male does. So based on that, we know therefore that she must have passed on her capital H, so we can see that the disease is present in heterozygotes because she is a heterozygote, and that only one allele is needed for a person to be affected by this awful disease. So we write here that the condition is present in the heterozygote. So only one allele is needed. And some children from affected and unaffected parents, as in the case of that first generation, are affected whilst some are not. A man has the same genotype as person 1 in the pedigree in diagram 1. Let's have a look at person 1. It's that person down here. We know they're unaffected, which means they must be lowercase h, lowercase h. Just making notes to myself. A woman has the same genotype as person 3. Let's look at person 3. Because person 3 is healthy, they must also be lowercase h, lowercase h. These two people are expecting a child. Calculate the probability that the child would be a boy, not showing the Huntington's condition, giving reasons for your answers. So we just need to draw out everything I was doing before. So we have a mother and a father. Phenotype. This will become all our reasons by laying it out like this. So we know they're healthy. We know their genotypes are this, which means their gametes must be this. We don't even really need to do a Punnett square because we can see that there'll be 100% this, which is healthy, which in probability terms means a 1.0. And then this thing about having a boy, remember there's a 0.5 chance of having a boy, 0.5 chance of having a girl. So because it's probability and you need both to be true, that they're healthy and a boy, you multiply the two together based on maths probability, and the answer here is 0.5. A man has the same genotype as person 2. Right, so we are looking at this person down here. We need to work backwards. We know that this man up here must be lowercase h, lowercase h. This girl down here must be lowercase h, which means this woman here must be capital H, lowercase h, in order to pass on the healthy allele to this girl, but the unhealthy allele to this boy. So I can immediately discern that that is number 2's genotype, so we'll steal that. And then what about the woman that has the same genotype as person 3? I've already worked that out, it's lowercase h, lowercase h. Explain the possible phenotypes and genotypes with respect to the Huntington's condition of any offspring they produce. So again, we can use our layout. So we're going to have man, woman, phenotype, genotype, gametes. So we know that this person here has the disease. This person is healthy. Sort out your gametes. Draw your Punnett square. So there's offspring 1. So we can see that there's a 50% chance of being healthy. Make sure you list that genotype. 50% chance of having the disease. The disease topic. A pathogen is a microorganism which causes disease. 2 types of disease include communicable diseases and non-communicable diseases. Now, communicable diseases may be transmitted from one person to another, and they are caused by pathogens. Pathogens include microorganisms like bacteria, viruses, and fungi. Non-communicable diseases cannot be transmitted from one person to another, and they are not caused by pathogens. There are various stages involved in the course of a disease. First step is infection. This is when the pathogen enters the host. The second stage is incubation, when the pathogen reproduces inside the host organism. And then the third stage is symptoms, which is when the pathogen damages cells and produces toxins, which is responsible for making the individual feel unwell. Why don't you tend to feel the symptoms of a pathogen as soon as they enter your body? Well, first up, only few pathogens enter in the first place. It takes time for them to reproduce, known as the incubation time. And it's only when you have large numbers of pathogens that you can really feel the symptoms of the disease they cause. I've already said that a virus is an example of a pathogen. Remember, viruses are non-living, they don't obey Mrs. H. Gren, they don't respire, they don't carry out homeostasis, they're not sensitive, etc. But if you think about the structure of a virus, it consists of a protein coat which surrounds either RNA or DNA. The protein coat sometimes has protein spikes on the outside of it, and enzymes are present such as reverse transcriptase, which is present in the HIV virus that causes DNA to be made from RNA. Another category of virus is the RNA virus. This consists of RNA, it contains no DNA whatsoever. Remember that viruses are known as parasites. This is because they infect a host and always cause harm to them. It's much more difficult to study viruses compared with bacteria, simply because viruses are much smaller and have far less complex machinery compared with a bacterial cell. The fact that a virus always has to reproduce inside a host also makes it difficult to study them. So we've said that viruses don't obey Mrs. H. Gren. Remember to watch the start of my all-in-one video if you're not sure what I mean by that. So how do viruses replicate? Well, first of all, viruses must infect the host organism. The virus particles attach to a host cell and inject either their RNA or DNA into the host cell. That will depend on the type of virus they are. The virus RNA or DNA is incorporated into the host's own DNA, and as the host replicates its own DNA, it replicates the viral RNA or DNA. Protein synthesis occurs. That is required in order to make more protein coats for the viruses. Lots of new viruses are produced. There's lysis of the host cell, which means that it bursts open and viral particles are released into the rest of the body. Lots of diseases are caused by viruses. Examples include polio, measles, mumps and rubella, HIV, which stands for Human Immunodeficiency Virus. This causes the disease AIDS, as well as the common cold. Ebola is also caused by a virus. If we turn our attention to Ebola now, let's talk about the various ways in which it may be transmitted. It's through bodily fluids, so that could be saliva, blood, semen and feces. If you come into contact with any of these bodily fluids that contain Ebola, you may get the disease. Various symptoms occur when you're infected with Ebola, including diarrhea, muscle pain, internal and external bleeding, and that's one of the horrific symptoms of Ebola is bleeding from your orifices, so that could be your eyes, your ears, your anus. It's very difficult to prevent the spread of Ebola. One way includes using disinfectant to clean surfaces, to avoid contact with an infected person. If the person dies, it's to ensure safe burial, isolated burial so people can't come in contact, and to wear protective goggles and clothing. Unfortunately, there's no effective vaccine against Ebola. Oral rehydration therapy may assist with the symptoms. The reason for this is because a person suffering from Ebola does get diarrhea and vomiting, so oral rehydration therapy can help replace any lost fluids and salts. The reservoir of a pathogen is an organism which carries the pathogen without suffering any symptoms. A good example is bats, which are a reservoir of the Ebola virus. So they carry the Ebola virus without suffering any ill effects. More detail now on HIV, which I've told you stands for Human Immunodeficiency Virus. Later on, this can cause the disease AIDS, which stands for Acquired Immune Deficiency Syndrome. So what happens with HIV is the HIV virus infects lymphocytes. The virus itself contains reverse transcriptase. So although the virus contains RNA as opposed to DNA, the reverse transcriptase enzymes causes the host cell to make viral DNA from its RNA. The type of cell infected is known as a lymphocyte. Remember, this forms part of the body's immune response to kill pathogens, and the specific type of lymphocyte infected is known as T helper cells. When the reverse transcriptase causes viral DNA to be made from RNA, that DNA is incorporated into the T helper cell's DNA. The HIV virus then instructs the T helper cell to make HIV proteins, and then as a result, the HIV proteins and RNA are assembled into more virus particles that when the cell undergoes lysis, it bursts open and releases them into the bloodstream. So how may HIV be transmitted? Well, through bodily fluids, so unprotected sex, the transfer of semen, using dirty, unclean needles, as the blood harbours HIV virus, and if you're really unlucky, infected blood transfusions. Initially, the symptoms are very mild, they're flu-like. It takes quite a large number of years for the disease AIDS to develop. The real issue with AIDS is because the virus infects T helper cells, which are lymphocytes. Remember I said that forms part of the body's response against pathogens. So it means that if you get infected with the common cold, your body is less able to fight it. And that's what actually causes the death of AIDS patients, is the buildup of all these other diseases which can't be fought against effectively. To avoid the transmission of HIV, condoms should be worn, clean needles should be used, and blood transfusions must be screened to make sure they don't contain any pathogens. Antiviral drugs can be used to prevent transmission of HIV and to slow the development of AIDS. There is no current vaccine available. Moving on to a different type of pathogen now, the bacterial cell. Remember, these are prokaryotic because they do not contain a distinct nucleus. Instead, they contain a nucleoid which contains the cell's DNA. They also have small circles of DNA known as plasmids. Bacterial cells are much larger than viruses They have cytoplasm, which contains water and dissolved substances. They have a cell membrane that allows the entry and exit of substances. They have a cell wall made of peptidoglycan. They have a slime capsule which helps protect the cell. Ribosomes enable protein synthesis, and a flagellum is a small tail used to help the bacterial cell move. Now, bacterial cells use binary fission to reproduce. They can't use mitosis. The reason for that is because they have no nucleus. So how does binary fission work? Well, the bacterial plasmids and nucleoids replicate. The cell membrane grows between the DNA copies. The cytoplasm divides and new cell walls form. And two daughter cells are formed, each with nucleoid apiece as well as many new plasmids. Now, in terms of how bacteria reproduce and how they grow, they'll follow a specific bacterial growth curve, which starts with the lag phase. This is when there are small numbers of bacterial cells present, due to the bacteria adjusting to their new environment, so you'll see a slow rate of growth. The growth phase, known as the exponential phase, you'll see a rapid increase in bacterial numbers, as they are now used to their new environment and they have a plentiful supply of oxygen and nutrients. Then the stationary phase occurs when amount of replication equals the death rate of the bacteria. And then finally the death phase, where bacterial numbers rapidly drop off due to a buildup of toxins and the fact that they're running out of oxygen and nutrients. You see mass death of the bacteria in the death phase. You may be aware that antibiotics are only effective against bacteria. They don't work against viruses, and that's because of the way in which the antibiotics work. Now, antibiotics work on the bacteria cell wall. They cause it to burst open by osmosis. Because the viruses have a protein coat, antibiotics are ineffective against them. It's possible to discern how effective antibiotics are by using a petri dish. So in this case, you have an aseptic technique, which means it will be sterilized. There'll be no bacteria present. You then use a swab or an inoculating loop to swab the agar jelly with your bacterial sample. You place your antibiotic discs on the petri dish, and then you seal your dish with tape. The antibiotics, which are most effective against the bacteria, will see the largest clearance zone around them, and you can actually measure that with a ruler, find out the diameter of the clearance zone, and use pi(r²) to work out the zone of inhibition. The larger the zone of inhibition, the more effective the antibiotic, because it's killed all the bacteria present. The agar jelly provides nutrients. You need sterile conditions to prevent the growth of pathogenic bacteria, so bacteria that you don't want present in your experiment. You're going to incubate your petri dish at a suitable temperature, such as 25 degrees Celsius, which enables your bacterial swab to replicate nice and quickly. A very nasty disease now, cholera, caused by the bacterium Vibrio cholerae. How cholera is spread is through infected feces. So someone suffering from the disease poops and their feces go into water, and then if you drink any water with those feces in it, you are likely to get cholera. The problem with cholera is that it causes super intense diarrhea, to the point where you produce stools called rice water stools. They're very characteristic, white and cloudy in colour. The person becomes massively dehydrated, and that's actually what causes their death. To avoid getting cholera, you obviously need to be drinking clean water which doesn't contain the bacterium. Good sanitation methods need to be used where that sewage is appropriately treated, and you need to wash your hands after using the bathroom. Treatment options include antibiotics used to kill the bacteria and oral rehydration therapy used to replace lost salts and water from the chronic diarrhea. Gonorrhea is an STI, a sexually transmitted infection. It's caused by a bacteria and it's spread through unprotected sex, so a good way to avoid getting gonorrhea is by using condoms, which provide a barrier. Symptoms include discharge from the penis whilst urinating, and if it gets so bad, it can actually lead to infertility. Anyone suffering from gonorrhea is often given antibiotics, seeing as that it's a bacterial disease. Athlete's foot is caused by a fungus. This is transmitted on wet changing rooms, so you'll find that people who swim a lot might suffer from athlete's foot more than most. You end up with sore red patches, usually found on your feet. That's due to your feet coming into contact with those wet changing room floors. To avoid athlete's foot, changing rooms should be dry. You should avoid walking with your bare feet on them. Keep your toenails short and use antifungal powders and creams in order to treat the disease. Because athlete's foot is caused by a fungi, antibiotics are not an effective treatment option here. We're now moving on to malaria. Malaria is caused by a protoctist called Plasmodium. It involves a vector. The word vector is used to describe anything used to transfer biological material, such as the bacterial plasmid used in genetic engineering to manufacture insulin, such as in the disease malaria. The mosquito is the vector because it transmits the protoctis, the plasmodium, to the host, the human. Malaria is a dreadful disease. It causes millions of deaths. Even if it doesn't kill you, it causes such long-term illness that people are unable to work, which really affects the economy. Most of the avoidance of malaria comes from making sure the mosquito doesn't bite you, doesn't infect you with the plasmodium. So how can you avoid that? Through use of mosquito repellent sprays, by wearing clothing to avoid the bites, sleeping under mosquito nets at night, again to avoid bites. You want to drain any stagnant water because this is a place where mosquitoes will reproduce. Insecticides may be used to kill the mosquito, and anti-malarial tablets may be used to kill the protist itself. There's no current vaccine for malaria, although anti-malarial drugs may be used to prevent the pathogen reproducing within the body. Another option is using sterile mosquitoes. These are mosquitoes which are unable to reproduce, therefore preventing the spread of malaria in this way, because there are fewer mosquitoes to bite people and pass on the plasmodium. Typhoid, another waterborne disease, is caused by a bacterial cell. Again, it's spread in the feces of infected people, which enters the water supply. To avoid the spread, clean water should be drunk, hands should be washed after using the bathroom, and there's actually an effective vaccine which stops you getting infected with the typhoid bacterium. For people suffering from typhoid, oral rehydration therapy may be used as well as antibiotics due to it being caused by a bacterium. Houseflies have got a bad name for themselves in the spread of typhoid because they land on the feces of an infected person. They pick up that typhoid bacterium on their feet and then they land on food being left out on the side, transmitting the bacterium in this way. Therefore, food should be covered. So the bacterial cell, the virus, the protoctist has entered our bodies. How does our body act to fight against them? Well, first of all, the lymphocytes detect the antigens on the pathogen. They produce antibodies to help kill the pathogen. Some of the lymphocytes turn into memory cells. This means that upon re-entry of that pathogen, the memory cells can produce lymphocytes in a huge quantity, meaning that antibodies can be produced faster, sooner, and in greater quantity in order to destroy the pathogen before it takes hold. Antibodies are highly specific. That's because they have a variable region on them which binds directly to the antigen on the pathogen. The variable region is complementary in shape to the antigen. Through binding of the antibodies, it causes the pathogen to stick together, making it more easily recognizable by phagocytes, which then secrete digestive enzymes and engulf the pathogen. The antibodies also produce antitoxins against the toxins produced by the pathogen, and they can even cause the pathogen to burst open. If you want a more active approach to avoid getting ill in the first place, you might choose to have a vaccine. A vaccine is an injection containing a dead or weakened or attenuated form of the pathogen. What happens when that pathogen enters the body, it doesn't make you sick because it's dead or weakened, but your antibodies respond to the antigens on the pathogen. The lymphocytes produce memory cells so that upon re-entry of that pathogen, antibodies can be made again much sooner, much faster, and in much greater quantity. So vaccines are an excellent way of protecting yourself against a particular disease. Herd immunity is all about giving a large number of the population a particular vaccine. This reduces the number of potential hosts that can be infected by a pathogen, reducing the chance of this pathogen being transmitted. There's far less chance of a non-vaccinated person coming in contact with the particular pathogen when herd immunity has been used effectively. Talking specifically about vaccines, so I already said some vaccines contain a dead version of the pathogen, such as in the case of whooping cough. Some contain a weakened form of the pathogen, such as the case of TB and measles, whilst other vaccines simply contain the antigens of the pathogen, such as the case of the influenza vaccine. Now to discuss the difference between active immunity and passive immunity. Active immunity is when our body makes the antibodies itself, and that comes about due to infection by a pathogen or due to a vaccine. But either way, your lymphocytes will respond to the antigens present on the pathogen. They'll produce memory cells which, upon re-entry of that pathogen, make a huge number of antibodies super quickly. This is called active immunity because it's our bodies producing the antibodies. A second type of immunity is passive immunity. This is different. This is when antibodies are transferred into our body It could be through breast milk when a baby breastfeeds, antibodies passed from the mother to the baby. It could even be while the mother is pregnant. Remember, the placenta transfers antibodies from the mother's blood to the baby's blood, giving them protection from pathogens. But the key thing with passive immunity is that no antibodies are made by the person. And as a result, passive immunity gives you only short-term immunity as opposed to active immunity, which gives you long-term immunity. We now need to distinguish between artificial and natural immunity. So artificial, as the name suggests, is really vaccines. It's when you inject that weakened, dead form of the pathogen into your body, which causes the response, which I've mentioned many times now. Natural immunity occurs when a person has become naturally infected with a particular pathogen, when they've been exposed, for example, to the TB bacterium. But as with both, the pathogen will be recognized by the lymphocytes, memory cells will be made, large numbers of antibodies will be produced upon re-entry of the particular pathogen. Let's consider how someone suffering from Ebola may be treated. Let's take a person who has successfully recovered from Ebola. This means that their blood will naturally contain memory cells that recognize the Ebola virus. What happens then if you give a person who's suffering from Ebola blood from the person that's recovered? It means that the blood they receive contains memory cells which will respond to Ebola. Those memory cells will produce lymphocytes which make loads and loads of antibodies which will attack the Ebola virus in the person suffering, therefore killing that Ebola virus. This is a much quicker way of fighting the Ebola virus. It doesn't require the person's body to have to take the time to produce the appropriate antibodies, and this is currently a more effective treatment option than taking medicine. So as I've already said, antibiotics are highly effective against bacteria because they cause damage to the bacteria cell wall, which causes the cell to burst open through the movement of water by osmosis. But where do we get our antibiotics from? Well, two places really: fungi and other bacteria. Unfortunately, we're finding increasingly that the antibiotics we use to treat bacterial diseases are no longer effective. They are no longer killing the diseases of old. But why is that? It's because the bacteria are mutating. This is an example of natural selection, of course, meaning that some bacteria survive, they're no longer killed by the antibiotic, and because they survive, they reproduce, and before long, the whole bacterial population is now resistant to a particular antibiotic. The reasons why antibiotic resistance is on the increase is for the following reasons. People aren't completing their course, the antibiotics are being over-prescribed, particularly in agriculture. You find that antibiotics are given to animals even when they're not sick. And antibiotics are being given in cases when they're not even supposed to be used. Remember, I said that antibiotics are ineffective against viruses. One such antibiotic resistance case which has been in the news a lot is MRSA. Now, MRSA stands for methicillin-resistant Staphylococcus aureus. Staphylococcus aureus is the name of a bacteria which usually doesn't cause that much harm, that much concern, and that's because the antibiotic methicillin has historically always been very good at killing this particular bacteria. However, due to all the reasons I've just outlined, we're finding that Staphylococcus aureus is no longer being killed by the methicillin, and this bacteria, which previously was fairly harmless, is now actually causing deaths of people in hospital. And the reason for that is because people in hospital often have operations, they have open wounds, and this is a place where that bacteria can enter the body and cause much more damage. The MRSA is very difficult to treat now. Many antibiotics are ineffective against it, so it's becoming a real problem in hospitals. So how can we prevent antibiotic resistance? Well, it's the sort of things I've just been discussing: only giving antibiotics when absolutely essential, making sure that patients complete the course of antibiotics. They shouldn't cease to take the antibiotics as soon as they start feeling better, because there might be some bacteria still surviving, and these ones are more likely to mutate and become resistant, so patients must finish their course of antibiotics. We should be avoiding using antibiotics in agriculture, particularly when it comes to giving them to healthy animals. And it's obvious that we need to have high hygiene levels in hospitals. As I've said repeatedly, antibiotics do not work against viruses. Obviously, we need to develop antivirals, but this is nowhere near as straightforward because the viruses infect our own cells, so it's hard to try and kill the virus without causing harm to the host cell. Decomposers are microorganisms such as bacteria and fungi which feed on dead materials, and decomposers have a huge role to play in treatment of sewage. Remember, sewage is human waste. The first step in sewage treatment is filtration. This is used to remove large particles. Sedimentation then occurs. This allows tiny particles to settle out. Aerobic bacteria and fungi oxidize any organic material. Anaerobic bacteria digest waste sludge. Methane gas is produced. Methane is an example of a biogas. Remaining leftovers can be used as fertilizer. And then chlorination is an essential process. This means adding chlorine to our water to make sure it's safe to drink, as it kills any pathogens present. So I've just described the treatment steps involved in a modern sewage treatment plant, but what about a pit latrine? As the name suggests, a pit latrine is a simple hole in the ground, which was used for hundreds, if not thousands of years previously. So a hole is dug in the ground over a sunken pit. Urine and feces drop into the hole and are broken down by microorganisms. Fly screens stop pesky flies reaching the feces and urine, and that therefore is important to reduce the transmission of disease, because remember I said if flies land on feces containing the cholera, typhoid bacteria, then they go and land on human food. This is a very easy way that typhoid and cholera may be transmitted. If sewage doesn't go after sewage treatment works and isn't properly handled, and say it washes into surrounding water sources like rivers and streams, what happens then is there's a huge increase in organic material in the rivers. Because the sewage contains a lot of nitrates, it actually causes algal bloom, so the algae in the water grow hugely. They block the light for the aquatic plants, meaning that the plants die due to a lack of oxygen. This provides food for bacteria, so the bacterial numbers increase, and because they aerobically respire, they use up all that oxygen in the water, meaning that it becomes anoxic, lacking in oxygen, so fish and other aquatic organisms can't survive. And remember as well that sewage contains an awful lot of bacteria in its own right, and this can actually increase the bacterial load so much in the rivers that that kills the fish directly. Right, that brings us to the end of this all-in-one video. I hope you found it super helpful. It was a huge undertaking for me, so please comment below if you found it helpful. Do give it a like. Remember, if you've enjoyed the wording, you like how concise and exam-driven my answers are, you can find all this and more in my online revision guide available to purchase on the website swhlearning.co.uk.